Hi, this is Ed Levin from Duke University, and I welcome you to our symposium, Marijuana Legalization, Brain Science, Community Impact, and Risk Prevention. This is brought to you by Together for Resilient Youth and Duke's Center on Addiction and Behavior Change. And first, I'd like to thank you for attending, and we'll be going from now until about 3 p.m. this afternoon. You can see on the screen here a list of very interesting speakers. And first, I wanted to give my thanks to Tyler Lee for providing logistic support for our organizational support for the program, and Nicole Schramm-Sapyta for helping put together the program, and most of all to Dr. Wanda Boone, who is our collaborator. She's the founder and director of Together for Resilient Youth. First, I just want to tell you a little bit about the Center on Addiction and Behavior Change. We're a center within the Duke Institute of Brain Sciences at Duke University. We bring together researchers, clinicians, community members interested and active in the treating drug abuse and drug addiction. And we have a seminar series. Actually, we have one next week. On Wednesday, Dr. Linda Perino will be speaking. Feel free to go to our website. If you just search for Duke and Center on Addiction and Behavior Change, you'll come to the page for the link. And now it's my privilege to introduce Dr. Wanda Boone, who is our collaborator for this program and the founder and director of Together for Resilient Youth. Wanda?
Hi, and good morning, everyone. I'm really excited to be here with you today. Together for Resilient Youth, we're in our 22nd year. It's so hard to imagine that that's the case. We're in our 21st year of our annual conference, which this is. So this celebrates our organization and our annual conference. And I'm so pleased to have Duke's Center on Addiction and Behavior Change to be a partner with us for the second year. And we look forward to next year and many years to come. Together for Resilient Youth is a coalition of coalitions. And in our coalitions, we have several sectors that are indicated. We have LIFT, which is our youth coalition, College Tri, which is our college age coalition. We have 550 businesses that have signed on with us. Our mission is to prevent substance use and the challenges associated with trauma so that we can get to the foundation of what's really the issue. And we'll talk about that later this afternoon. We have faith-based law enforcement, community members from all of the sectors in Durham, all of the districts in Durham, I should say. We have health representatives, schools, and sadly, parents that have lost children due to overdose, and parents in general. We have many, many, many partners, not only the businesses that I spoke of, but also the school system and North Carolina Institutes of Medicine that I was a part of to help create the document, Healthy North Carolina 2030. We have other coalitions that are a part of our organization across North Carolina. We're based in Durham, and across the United States. And just recently, internationally, including Australia. So we're really, really excited about the prevention message, focusing on trauma. And we invite anyone and everyone to our monthly meetings. And then we have other meetings scattered throughout the year. So thank you so much for being here. We're excited that you're here. Lots of information that we have to give you. And so we will continue with our program, Ed, unless you have something else. Are we ready to start?
Yes, we're ready to roll.
Ready to roll. So first up, we're excited to have Dr. Jonathan Livingston and Crystal Taylor from North Carolina Central University. I'm excited to present what they have done, their work over several years, docudramas. We need innovative ways in order to prevent substance use, particularly when we're talking about underserved populations. The social determinants of health are in top of mind for everything that we do at TRI, and I'm sure it's something that you've been talking about too. So docudramas are a way to, and she may use another term, I'm sorry, it's a way to identify whatever the subject is that we want to prevent, but put it in such a way that it is palatable and culturally appropriate for everyone, but in particular communities of color. So Crystal, please take it away and do a little bit more of an introduction than I did.
Good morning, everybody. My name is Crystal Taylor. Thank you, Dr. Boone. It is a privilege for me to be able to be here today to share with you guys the work that we've been able to be doing with North Carolina Central University and my nonprofit organization, Get Happy. So I'm going to talk to you a little bit about the ethnodramas that we've called them, but they are technically mini series and films that we've been able to create. First of all, I just want to share a little bit about what Get Happy does and what we're able to be able to do with colleagues and different organizations. Our mission is to restore the quality of life for people of color and to be able to do that innovatively through health and wellness, innovative education, and community engagement. And so when we found that before the pandemic had started, we saw that there was a decrease in family gathering around TV and entertainment. And so one of the things that the ethnodramas do is to define written transformation and adaptation of research that we take. And so with that, we've been able to create transcripts and get observations from participants to find out what information is needed to be able to educate the community. So right now we are being able to talk around breast cancer in young women, prostate cancer, and HIV, as well as cervical cancer and opioids. So this coming year, we were able to research a demographic, which was at a facility that these individuals live in that are actually going through, they're going through care there for opioid abuse, and get information on what really happens in their life, a true story that we can be able to relate to and tell. And so with this information, we have a playwright that comes in, he writes a script, and we create a whole series that was able to educate people on what the true life and struggle is of a person that's going through addiction, HIV, or breast cancer. There's 3 specific aims that we try to touch when we create these films. The effectiveness of this is really important to make sure it increases the knowledge around the disparity or the awareness of the disease, to be able to help foster behavior in the community, so that way people will be engaged more and more aware of what's going on. We also test the hypothesis around the drama-based health education. So what that means is making sure we create platforms on social media, and the tools that we use through that are social media, delivering health education to communities, because social media has driven the engagement with teens and families, has driven since the pandemic has started. And even before then, it was a great gateway to be able to communicate with people at the level that they are in. And the third aim is to be able to provide experiences and education for students. This enables them to be able to connect with communities that are struggling with these disparities and their diseases, to be able to meet them where they are and be able to communicate with them the needs that they would be able to provide in their prospective education and careers. So as I mentioned before, some of the films that we've been able to do: cervical cancer, COVID, also responding to COVID and understanding COVID, opioids, diabetes, and we're going to be working on cardiovascular health disease in the future. And so the method that we use is just like creating a regular television show. We have a playwright. We have a playwright that creates a script. Excuse me, y'all. Excuse me one moment.
While we're waiting for Crystal to take charge of everyday life in Zoomville, I want to acknowledge some of our dignitaries that are here. And so let me make sure that I don't leave anyone out. Crystal will get back with you in just 1 second, if you don't mind. So we have Commissioner Wendy Jacobs here. So Wendy, if you would like to say something, I believe you can unmute and do so. And so please do if you'd like to make some remarks.
Good morning.
Good morning, we can hear you.
Good morning, Dr. Boone and everybody. Thank you so much for giving me the opportunity just to bring greetings. And I just want to thank you, everyone who has helped organize this incredible symposium today. And I just appreciate all that you bring to our community, Dr. Boone, with all the coalitions you're involved with, and also the Duke Clinic for the important research that you're doing. And I'm here to listen and learn, and I'm excited to hear what Crystal is sharing with us right now. So thank you so much. And I'm just going to sign off and participate for the rest of the day. Thank you.
Thank you so much. And Deidreana Freeman. And while that's working, we have Steve Mange, I believe. Steve, if you would like to unmute and say something, then please do so. And then we'll get right back to Crystal and her excellent presentation. If not, we can save you for later on. Okay, so Crystal, sorry for that interruption. Glad you were able to take care of your beautiful son, Mansa, and please continue. Thank you so much.
Thank you, Dr. Boone. I apologize, you all. Thank you so much for your grace. But in continuing, the ethnodramas are so cool and unique because they're created just like a regular television show that we would see on TV. We build everything out from inception to production. And the great thing about what Get Happy has been able to do with Duke University School of Nursing, North Carolina Central University, is really learning about the disparity and the disease that people of color are struggling through, and learning what they didn't know prior to, the disease, in this case, opioids, and taking that information and using it to teach and to bring more awareness around the disease and the disparity to the viewer. We go as extreme as, having focus groups of individuals who do have those diseases or who have suffered, whether it be on the, intake level, or a family member being, having to go through things that they have, dealt with their members struggling with the disease or the disparity. So we have these sessions and we're able to gain so much feedback and awareness and understanding around what the struggle looks like in all different perceptions. The playwright is hired and he creates an amazing script that we then review as a team to make sure it's fitting all areas that can be touched that are relevant to healing and restoring quality of life. And so then we go out and we do what we do. We find actors, we do a casting call, we have a crew that we use shooting, we find sets and just build everything out. The script is adjusted to make sure that it's age appropriate for all family to be able to view, have discussions around it, and it's entertaining. We do rehearsals, we have a director, we have a stage manager, we have an executive producer, which I've been able to hold the role on in several films. And we shoot the story, and it just becomes so incredible for us, even as the actors and the production team, to be able to connect with the stories, to be able to learn information, have more awareness around what our community is suffering through, even if it's not our own personal story. The beautiful thing about the ethno dramas is that everyone has a story to tell. Everyone has an uncle, everyone has an aunt, a cousin, or themselves that can relate to one of the disparities and diseases that we've been talking about. And, statistics have shown 30 to 40% of families were not engaging together in the setting in the living room, watching TV, having discussions and learning about disparities and having more awareness. What the pandemic has done for families is brought them closer together. It's made dinner time at the table, watching TV or watching your family sitcom more available and more attainable for families, which then creates a great opportunity to have more discussion, awareness, and openness around things that people are going through, struggles that they're seeing, different things, and also a time for healing. So with the ethno dramas, it was our intent to make sure it was intentional. It was bringing awareness and knowledge around what people are going through, what they need, and how we can meet them to serve them. Before we release the films or show them to the audience, we do a survey with individuals about their knowledge and awareness on the topic. We do the survey to the viewers. We make sure they answer all the questions, and that helps us build an awareness of what people did not know going into watching the films. The production is shown, is performed in front of audiences or live viewing, and after we have a panel conversation to talk about what people saw, what they felt, and what they learned. These conversations, I have a marketing analytics background, and these conversations are incredible to hear because people really have been able to develop an openness with us to be able to share what they did not know and what they're happy that they did learn. And also they're excited about being able to know what they can do. The ethno dramas have done a great job of communicating with people in a way that makes them feel like somebody saw them, people are hearing them, and people are there to serve them. And it's just great. It's great film. It's great TV to watch. Everybody has that favorite sitcom or movie that they just always love watching because they can resonate with it so well. So the incredible thing about these films is that we've been able to do that every single time and just nail it for people in the community watching. After people watch the film, we survey them again, maybe 2 to 3 times, just depending on the demographic and at the time. And then we're able to take that feedback and use it to create engaging, innovative opportunities of education to be able to help people and serve them. And that's just one of the greatest benefits I think comes from this, being able to create, meet people where they are, where they've been, and where they want to go, and create visuals and productions around that to be able to help and engage with people. We have accomplished a lot in the last 3 years. When we first started, we didn't really know what we were getting into, what we were doing. And so we did not work as innovatively, I would say, than we do this year. But we've had over 300 people attend the plays between Charlotte, Asheville, and Durham. And we've been able to, have some great conversation and feedback. The statistics have increased significantly with the demographics that we've surveyed around their education, awareness, and willingness to be able to serve in the communities that people are suffering in around these diseases and disparities. One of the things that we've been able to do thus far is create 2 or 3 scripts now. Crisis of Moments is a COVID-19 play that was written by Desana Hanu and directed by Dr. Sabi at North Carolina Central University. And that was truly just a spinoff of watching Saturday Night Live, with all the content surrounding around fun tidbits or funny tidbits or educational tidbits around what COVID-19 is. Everything that we saw in the news, we used that to be able to dictate the complete truth around and factual information around what people need to know about COVID. And then we had some funny little dibs in there around, everybody knows that it's a serious matter when you see face masks blowing in the wind or on the sidewalk, and the poor little lonely face mask. So there's ways that we have been able to implement comedy in these, but also a very awakening, knowledgeable fact that people need to be aware of and really understand what's going on with these disparities. We also did a film right after Crisis of Moments to help engage people with what they can do around the pandemic, how they can survive the pandemic, if you will, in a notion of what people are needing and what they're feeling. So it was a spinoff of that, but it was also educational to understand, like, to make a decision around the vaccination. We used the setting of like a late night show with Jimmy Fallon to create that, and it was very well received. We were able to have people that were engaging guests, if you will, that were able to talk around, if the FDA or if they have zombies in the back that they're experimenting on, funny things that were able to get people to listen, but also be aware and want to get more educated. So we've been able to do that with these last films, and I'm very excited. We've created TED Talks with community leaders. Dr. Wanda Boone was a part of one of those conversations that we created around opioids. Being able to engage the community and the churches, TROSA, and other healthcare communities in the state has been pivotal to us being able to have the research and the demo to be able to talk to and create these panels. Get Happy is really proud of working with the Duke School of Nursing to create a production, if you will, that was centered around HIV and women's health. And that we created those literally in 4 episodes. That takes around 2 to 3 months to do, but we were able to do that within a month and a half. And with that, those films would be used to teach women in beauty salons around health equity and HIV and relationships. Creating images like this that people can see themselves. We all know that, on network TV, we don't see a lot of Black dramas and Black TV shows that really speak to. We tried to look at The Neighborhood that comes on CBS, to use that as a great filter to help educate people. Cedric the Entertainer does a great job of using comedy to educate people around, going to get his yearly checkup as a man, as a Black man, having the struggles to do that and be confident and share that information with his wife. Racism that goes on in the Black community that white Americans that are not necessarily immune to or understand. The kind of conversations that TV show showcases has been incredible knowledge for us to be able to use that in our ethnodramas. Right now, the ethnodrama is not necessarily available online because they're products of the university and do CTSI, but we've still been able to share them with community church groups, community organizations, and other nonprofits that are working directly with those demos that would be affected by it, that would need the support. So some of the takeaways that I could share with you all around the engagement levels, what we've seen with people watching and using ethnodramas to engage with their communities and their clients. There's been a 50% increase since the pandemic started, since the first year into the pandemic, with families coming together, sitting together and talking and engaging and having more awareness around what's going on individually and in their personal communities of their own peers. And what we've seen is that there's more engagement. Mothers and sons and fathers and sons are talking more, communicating more. They're sharing more information across their generational education limits. The intention is more, there's more intentional conversation, being that they are asking more questions around what they saw and what they were educated on and watching the videos and having more intimate conversations with their sons and daughters and with their peers. The education and awareness with the disease or the health disparity. That is a key point that people need to be aware of, that one of the main reasons why people aren't talking about the disparity or the disease is because they don't know anything about it. In our cervical cancer production, when we specifically said HPV, the communities they were confused and thought we meant HIV. So that was probably over 45% of people that did not know what HPV was that were attending that film, that watched. So it was a critical takeaway for us to recognize that the reason why communities of color are not talking about certain disparities or diseases is because they don't know anything about it. So that's why it was really important for us to make sure we put a lot of facts, a lot of information, a lot of just conversations and comedies around the disease, the disparity, and what that is and what it can do to you and how you can prevent ways to not get it. Once we implemented that, it made everything incredibly easy to showcase. And lastly, the acknowledgement of feelings. One of the things I know personally that struck me was that when meeting people that were going through their struggles with opioids, we did not meet the patient, but we also met their families. Meeting them and understanding how they process their feelings and how the client person struggling with the disease process their feelings was an incredible gateway to being able to communicate to families that are watching the films and people that are watching the films. People need to know how to meet people where they are, dealing with certain disparities and diseases that could be triggering for them emotionally and mentally. And so we were able to take that information, put it in the films, however we did, and create engagement that was really like breakthroughs for people to be able to communicate with one another. So that's the work we've been doing. I know that was a lot, and I'm excited about what we're going to be doing in the future with the university. And if anybody ever wants to reach out and connect with us, please feel free to reach me at gethappync.org or Crystal at theugretrokids.com. Thank you.
Oh, thank you very much, Crystal. That was an outstanding presentation. I look forward to seeing more of your productions. So anyway, I think that next we'll proceed on with Dr. Scott Parnell. We do have Dr. Jonathan Livingston who can present later in the day, but now let's go on with Dr. Scott Parnell, who is assistant professor in the Department of Cell Biology and Physiology, and is also a member of the Bowles Center of Alcohol Studies at University of North Carolina, Chapel Hill. And he's going to speak to us today about his work in laboratory research on neural mechanisms of cannabis's behavioral effects. Scott?
Can you hear me okay? Ed and Linda, or Ed, Ed and Wanda have tasked me with trying to at least give an overview of the mechanisms of how cannabis affects the brain, both in adults and I will also go into some of our recent research in embryonic development. So, we all are relatively familiar with the main components of cannabis, THC, which is the main psychoactive effect, and as well as CBD. And both of these have some potential medical uses that could be promising. But there are also some downsides to some of these compounds that I'll go into. So one of the interesting things about cannabinoids is that they signal through the distinct receptors, the CB1 and CB2 receptors, and I'll describe a little bit more about them. But these cannabinoid receptors are present throughout almost the entire body. But the ones I'll focus on today that I've been tasked with sharing are the ones in the brain, as well as the newfound ones in the embryo. And so these are probably, at least to me, two of the most important ones because they affect how we think, how we act, but they also affect how we develop. So when we look at the brain, so this is an adult human brain, and what this is looking at, this is a PET scan. So this is a slightly radioactive CB1 agonist, so it binds to CB1 receptors in the brain. And so it sort of lights up everywhere that we see these receptors in the brain. And you can see that these CB1 receptors that bind cannabinoids like THC are present throughout the entire brain. So you can imagine that when we consume cannabis products or smoke marijuana, et cetera, you may be affecting all sorts of different parts of the brain, such as the caudate-putamen, which affects movement and reward, the cortex, which affects sensation and perception, the pituitary down here, which affects hormones and stress response, et cetera. So this is mostly just to show you that these receptors are rather ubiquitously expressed throughout the brain. And so it, of course, can have a lot of different effects. Now, unfortunately, cannabinoid receptor signaling is actually really complex. And as I hope to show you throughout this brief presentation, we actually really have a lot to learn, even as much research has been done over the years. But we know that cannabinoids, whether they be the cannabis product delta-9 THC, which binds to the CB1 receptor, or even the synthetic cannabinoids that are often found in products like spice and K2, which are really, really potent. They all bind to these cannabinoid receptors with different efficacy. And these cannabinoid receptors, when they bind one of these products, whether it's THC or the synthetic cannabinoids, they are coupled to these G protein receptors. And these G proteins are second messengers. So they are released by the receptor, they go into the cell, and they affect the downstream signaling. And that's basically how these receptors work. So basically, the ligand binds to this receptor up here in the pocket, and it releases a G protein, which goes on to signal within the cell. And we know what these receptors are doing best in the brain because that's where it's been studied the most. So if you look at two neurons here, this is a rather simplistic diagram of two neurons. So you have this neuron that's signaling to this one through normal neurotransmitter release. And it doesn't really matter which neurotransmitter it is. But what happens is that this cell signals to this cell, and then this cell signals on downstream. But what happens in normal situations is that when these receptors are activated, you release an anandamide and 2-AG. These are endocannabinoids. So these are normal endogenous cannabinoids that are present in our bodies at all times. And what they do is they feed back onto the cell the presynaptic neuron to sort of limit this activation so you don't get overactivation. So you can imagine that then if you were to consume THC, that you could then get an abnormal effect on the presynaptic neuron that then affects the postsynaptic neuron and it affects signaling. That's a rather brief overview, but that is basically how one of the ways that cannabinoids work. Now, unfortunately, it's not quite that simple all the time, of course. And the more we learn about the cannabinoid system and the endocannabinoid system, the more we're starting to realize that there are other players involved here. So here again is that presynaptic neuron. It's firing, releasing neurotransmitters that go to the postsynaptic neuron to propagate signaling throughout our brains, throughout our body. But neurons and their extracellular matrix also regulate themselves. So you have a somatodendritic mechanism, whereas the cell can auto-regulate itself, again, through endocannabinoids. So that's another way for neurons to regulate themselves. You also have non-neuronal cells called astrocytes out here in the periphery that are also regulating how these neurons are firing. So again, these astrocytes, through various stimuli, can release these endocannabinoids, which also can regulate the presynaptic neuron. And then, of course, you have that postsynaptic function that I talked about a minute ago. So you can see that we have lots of different ways that cannabinoids can control neuronal firing and thus control behavior and normal function. And of course, not only does it affect acute function and acute neuronal firing, there are also these long-term effects to gene expression and intracellular signaling that are happening as a part of normal cannabinoid function, but which of course can be hijacked by the use of cannabis products. And in some cases, these are good. For example, CBD has been approved by the FDA to control uncontrollable epilepsy, for example, and especially in kids. And it seems to work quite well. And it works by getting these mechanisms under control. But of course, there is also the potential for abuse. And one of the ways that is the downstream effect of, and one of the reasons why so many people use marijuana, is that a lot of these mechanisms of action is to enhance dopamine activity. It is this dopamine activity that is controlled by GABA and glutamate, and very, very briefly, that is sort of associated with the reward and the pleasure effects of cannabis. But what has been recently shown is that this is also another way that that the cannabinoid system is complicated. I mentioned that CB1 here is a G protein coupled receptor, so it signals through downstream small molecules. But what the dopamine receptor, which we have here, is also a G protein coupled receptor. And these dopamine receptors can bind with each other and alter how they signal. But what we've recently realized is that CB1 and dopamine can also bind to each other and affect how they signal downstream. And so I just show this to point out the really complicated ways in which the endocannabinoid system is designed within the brain. It's really different brain regions have different sets of these receptors. They have different sets of extracellular milieu. And what that means is that we don't know nearly as much about CB1 or cannabinoid effects as we should. We really need to know a lot more. Also complicating all of this is the fact that over the last 20, 30 years, the concentrations of THC and marijuana have dramatically increased. So marijuana is like any other drug. There's a dose-dependent effect. And so a lot of what we may have studied back in the 80s and 90s, back here at what we consider to be relevant concentrations of THC, may not really be relevant research or nearly as relevant as we thought anymore, simply because our concentrations of THC have gone up so much. And at the same time, some of the other cannabinoids within marijuana, like CBD, actually have gone down over the years in normal marijuana. Now, CBD is still important because, as we know, you can go to any convenience store and buy CBD oils. They're used quite a bit, and they actually seem to have, at least anecdotally, some decent efficacious effects. But in addition to this increase in THC within most marijuana products, we also, of course, have the fact that we have, as a country, have increasingly legalized the use of marijuana. And so that of course means that a lot more people are using marijuana than were 10 to 20 years ago. And in addition to us as a whole using more marijuana, there was a paper published a few years ago showing that even among pregnant women, there is a dramatic increase in the use of marijuana. And so this was a California cohort of pregnant women. And they broke it out by age. So under 18 and 18 to 24 were the main users. So it went from 10 to 15% to almost up to close to 20% in these populations. So there's a large portion of pregnant people who are using marijuana. So that is yet another area that we don't know much about. We don't know what the effects of marijuana are on adult populations, let alone what it might be doing during development. And so a couple of years ago, my lab started looking at the synthetic cannabinoids. So these were these synthetic cannabinoids are really potent analogs of THC, the natural product in cannabis. And sometimes they're 50 to 100 times more potent. And for a while there, they were really highly abused. Their use has started to teeter off a bit now that we're legalizing natural marijuana. But their abuse really resulted in a lot of emergency room visits just because they were so potent. But we wanted to look and see what effect these drugs might have on development. And so we used our mouse model. So we are normally a lab that studies fetal alcohol syndrome and the effects that alcohol has on early development. But we wanted to see what these cannabinoids might be having on early development. So we like I said, we study really early development. So if this is a mouse embryo during day 7, that's equivalent to day 17 of human gestation. Here it is slightly later. So this is eventually going to form the brain, this part up here. And so the brain is just starting to form at these early periods. So this is early 3rd and 4th weeks of human development, or equivalent to, so very, very early. But this is the point when we're really starting to develop our brain and our face. And so impacting these development during these periods can really have a long-lasting impact. So we gave an acute dose of this synthetic cannabinoid and looked to see what effect it had on the mouse during these early periods of development. And the great thing about the mice at this point is that with alcohol and other drugs, what we see in humans can really be nicely copied in mice. So mice and humans are developing very similar at these early stages of development. If you were to look at this human embryo, this picture of a human embryo, you could really hardly tell the difference between a mouse. So development that is occurring is very nicely conserved across species here. And so when we look at a mouse embryo right around birth, mice are born a little bit premature. We can see here a beautiful little mouse fetus with its little eyes. That here you can see a nice little nose and a nice little philtrum. So that philtrum is that spot just under your nose. And this is what a normal mouse looks like. You can see the ears are just starting to develop. Well, with just a single acute exposure to the synthetic cannabinoid, you can see that we have really severely impacted development. And this is even with the moderate doses of these drugs. So you can see here a smaller head indicating a smaller brain. We can see eyes that are too small, or in this case, almost absent. We've really altered that filtrum here, and we see a smaller jaw. Again, here's that small abnormal eye compared to this one. And what's really interesting about this is that these are actually some of the main effects that we see with fetal alcohol syndrome. It really seems to be phenocopying FAS. And when we look at the brain, which is really our main focus, we're really developmental neurobiologists. When we take a section through the front of the face or the brain, so here are the eyes, nostrils, this is the tongue, this is the palate, the roof of your mouth, and here's a section through a nice normal mouse brain at these stages. When we expose to these cannabinoids, we really see a lot of ventral midline effects. So we've really lost a lot of brain tissue here. If you look at this one, we've actually caused a cleft palate. So now we don't see many cleft palates with alcohol exposure. So these cannabinoids are doing something else in addition to. But these effects that we see in the brain are very, very similar to what we see with prenatal alcohol exposure. And when we look at the eyes, just because we can look at eyes pretty easily, and eyes are affected in a lot of different developmental disabilities. When we look at just the eyes, we can see that we get a really nice dose-dependent effect of this synthetic cannabinoid. And we're seeing effects even at really small doses. So it appears that when we expose the embryo at these very early critical stages of development, we really get long-lasting and enduring effects, even at small doses. Now, of course, we wanted to extend these findings to other compounds. So we looked at another synthetic cannabinoid, but we also looked at THC and cannabidiol, or CBD. And what we find is that THC also has the same effects. Now, these doses are a little higher because THC is not quite as potent. But what we find is that even with CBD, we still see effects. They're more subtle than what we find with THC, but they are certainly there. And I will point out that CBD is not a classic CB1 receptor agonist, but it is still impacting development somehow. And of course, being an alcohol lab, we had to try them together. Marijuana and alcohol are often used together, so it was natural for us to look at them. And so here we're looking at just alcohol alone and these two. So this is a moderate dose of alcohol, and it has a little bit of effect of development. Alcohol is like any other drug. It has a dose-dependent effects. When we give a slightly higher dose of alcohol, we then get a slightly greater effect. Now, over here, we have just the cannabinoid alone. Again, we're going back to the synthetic just for the sake of this experiment. But when we combine even a low dose of alcohol with the cannabinoid, we're getting this synergistic effect. And when we give a high dose of alcohol with that moderate dose of the cannabinoid, we affect almost 100% of the fetuses. So we're actually having almost a synergistic effect, where the two of these drugs together are not just additive, they're synergistic. So they're having a more than additive effect. And we find these effects that are consistent across any of the cannabinoids that we're looking at, including THC. Now, we've known for a long time that alcohol affects development during these periods of development by affecting the sonic catch-off pathway. And I won't go into too much what this pathway does. It's a cell biological pathway that is a growth factor. So here again, here's an embryo at these early stages. If we take a section right through the middle of this embryo, so you're looking at the back of the embryo, this is going to form the brain and the spinal cord. Take a section right through the middle. We have this green expression as sonic catch-off. it's a growth factor. It simply makes this part of the brain grow. And what we've known for quite a long time is that when we reduce this sonic hedgehog signaling, that signal that is so important for normal growth, we get abnormal brain and face development. We see that in a lot of genetic developmental disabilities, but we also see it with alcohol. And so here's a study published by a group many years ago, where again, you're looking at a midline section through this embryo. This is going to form the brain up in here, and then the spinal cord. This black is sonic hedgehog expression. And when after exposed to alcohol, you can see that it's greatly reduced. And so what that does is that you don't get the normal proliferation within the developing brain that you need for normal development. And so naturally, the next thing we did was to look and see what these cannabinoids are doing to sonic hedgehog expression. So here we get a decrease in sonic hedgehog expression compared to this control with alcohol, as shown by this group many years ago. And indeed, when we measure it, we're just looking at expression, we get a greatly decreased sonic hedgehog expression. We also, downstream of sonic hedgehog, this is sonic hedgehog activates this transcription factor, which turns on gene expression. We also get a decrease downstream of sonic hedgehog, as we would expect. So here we're showing that like alcohol, these cannabinoids are also decreasing sonic hedgehog expression and really impacting development through a very similar mechanism, we think. And just to kind of briefly show you here, when you have sonic hedgehog activates this receptor activates this other G protein coupled receptors called smoothened. So this is another G protein coupled receptor, just like CB1. And I'll ignore all of this, but just know that when this G protein coupled receptor is activated, this little G protein goes in and causes an increase in normal cell proliferation. And so that's how this growth factor pathway works. And so somehow alcohol, through some mechanism that we're not even really sure about, alcohol is decreasing this pathway. But through a whole series of mechanistic experiments, what we sort of discovered was that this CB1 receptor here is also present in the embryo, which we didn't know previous to these experiments, but it's also binding to this smoothen protein, this protein that you need to turn on for normal development. But this CB1 protein is binding to it, and when it's activated, it's actually inhibiting its function. At the same time, we and others have shown that these cannabinoids are also directly inhibiting the smoothen protein, which is again necessary for normal growth and development. And so we've kind of shown that this CB1, through its rather ubiquitous effects in different parts of development, different parts of the brain, in both adults and humans, is we can really have very profound and differing effects depending on what is being expressed along with it. And so I'll just briefly sum up that, that in the adults, these cannabinoids alter the multiple neurotransmitter systems through these GPCRs. So all of the cannabinoids that we've studied so far, including THC, CBD, etc., they all seem to affect both the CB1 receptors as well as other CB1 receptors or other GPCRs like smoothened, which is necessary for that normal growth factor pathway. And they seem to impact brain and face development, at least at these early periods. You'll learn about adolescent periods later on, but they really seem to affect normal development. And what is interesting, I think, is that given this similarity in what we see with cannabis products and alcohol, we have studied fetal alcohol syndrome for a long time. Maybe we have been ignoring the effects of cannabinoids during development too. And it's because there is this overlap in we need to do better at teasing out these different things. And we also need to be aware that these cannabinoids, while they have many positive effects, for example, again, children with intractable epilepsy or adults with, who are on cancer treatment, we do just need to be aware that these cannabinoids really may be impacting development, particularly at really vulnerable stages. And so with that, I will stop sharing and Wanda decide.
Yeah, thank you very much. And that was a great presentation. There is a question about could you offer an approximate dosage of the cannabinoids with your studies?
Absolutely. So we really tried to extend our dosages of cannabinoids to kind of be lower than what would be a normal exposure, and then all the way up to, of course, the really high dosages were high. We know that, probably higher than a lot of people would get without ODing on these cannabinoids. But in all of our studies, we tried to mimic what would be a relevant doses.
Okay, thanks. I had another question about, you saw that there was an interaction with ethanol, and what about other drugs that we've used, like tobacco or other substances that are abused?
That's a good question. We know that a lot of those impact development too. And we have not personally gotten to them simply because we've been preoccupied with just alcohol and cannabinoids. But they absolutely need to be studied along with opioids and everything else that we tend to use as humans.
And here's another question that's come in. So just overall, what do you think are the most important or devastating impacts of marijuana during development in particular?
That's a good question. So I think the most devastating impacts, at least as we have studied them so far, I think there's a lot more that we need to know. I think the most devastating impacts are on the brain, right? Mm-hmm. Much like alcohol. We are just starting to get into some of the long-term behavioral impacts. And at least our preliminary data seem to show that the long-term effects of even, a short-term exposure during early development tends to have an effect on learning, as well as possibly social skills.
Mm-hmm. Great. Okay. Well, that's not great, but your presentation was great. That's awful. But anyway, it's important to be aware of, and your research is vital for ascertaining the risks, and hopefully those will be attended to. But thanks for a great presentation. And now we're going to move on and another lab science presentation. Next, we're going to switch focus in terms of age, from adult and embryonic development to the critical period of development called adolescence. And so privileged to have Dr. Diana Dow-Edwards, who's professor in physiology and pharmacology at Downstate Medical Center in Brooklyn, New York, who's going to speak to us about adolescent vulnerability to cannabis effects on neurobehavioral mechanisms. Diana.
Yeah. How about the mouse? Yes. Okay, great. Hi. Yeah, thanks so much. I really appreciate the opportunity to go through some of this information with you all. And it sounds like you have a great group down there, and I'm really happy to be a part of it. When Ed first asked me to do this months ago, I'll pick up the recent literature. And when I actually sat down to look at the recent literature, there have been more than 2,000 papers just on marijuana and adolescence in the last 2 years. So I think with the pandemic, everybody seems to, since they can't go to the lab, they're going to stay home and write papers. And we've had some incredible, really fantastic reviews that have come out in the last 2 years. So I'm really happy I've been able to look at them. So Okay, so Scott really helped me out with the pharmacology here, so I don't have to spend too much time on it. He talked a lot about the THC CBD issues, of course, and also mentioned that the potency is increased. What I just wanted to just mention here is that cannabis oil, which is used in vaping, often exceeds 70% THC. And then there are some extracts called wax or shatter or solids that are produced by regular marijuana, but the THC content can actually exceed 90%. The national average was about 56%, and in Washington State, it was up over 70%. So the concentration of THC is directly proportional to the damage that's produced, and basically, and there's a dose-response relationship. And so I'll try to hit on that all throughout my presentation. But this is an illustration of recent years, 2015 to 2018, where USA 12th graders they're self-reporting whether they smoked marijuana, which went down, whether they were using edibles, which went up, or vaping. And as you can see that the vaping went up, and this is the big problem. And this increase in vaping with up to 70 or more percent THC in these liquids that these kids use, this is really serious exposure. Scott did a great job, talked all about the pharmacology at the nerve terminal. Basically, it's the same story here. The only thing I want to just emphasize here is a little more on the presynaptic site. While yes, the postsynaptic, the presynaptic inhibition of neurotransmitter release, as Scott beautifully described, is the main action of the endocannabinoids in the brain. But there's also at the postsynaptic site, when the either GABA or the glutamate interacts with the postsynaptic receptor, there's also the stimulation of the production of anandamide, which is one of the endogenous cannabinoids in the brain. And this anandamide, through the TERP V1 receptor and through the CB1 receptor on the postsynaptic site, tonically inhibits and alters firing of that postsynaptic element. So there's both presynaptic and postsynaptic actions of the cannabinoids. And here, I just want to emphasize, I'm always interested in sex differences, that this postsynaptic issue is much more important. First of all, there are big differences between males and females in terms of metabolism. Liver females metabolizes marijuana into many more active constituents compared to male. And in the male, marijuana, THC, is stored in the adipose tissue. There are big sex differences in the endocannabinoid system itself, as well as in the hypothalamic pituitary gonadal axis. Now, in females, that presynaptic terminal effect of the endocannabinoids is relatively more important, while in males, it's that postsynaptic constituents that is relatively more important. Both, of course, occur in both males and females, but many of the postsynaptic effects are responsible for what we see happening in males. Now I want to talk about the development of the endocannabinoid system in adolescence, and I'm sure most of you have seen this graph from 1993 many times, so I won't spend too much time on it. But you have, of course, in the limbic system, in the striatal system, or in the mesencephalic system, you have from days 10 to day 70 in a rat. You have the increase and then a drop off to adult levels in the CB1 receptors, the cannabinoid receptors. And at approximately 30 days of age, the open bars are the males, and these are the females. Both genders, both sexes, show peak CB1 receptors in the limbic system. However, in the striatum and in the mesencephalic area, that's your ventral tegmental area, we see a difference. Females peak at 30 days in both, while males peak at 40 days. And this is undoubtedly related to the delayed puberty, which occurs in males compared to females. Now, why is this important? Well, it looks like this exuberant projection that is happening in adolescence, which is a normal process, in adolescence, there are cannabinoid receptors on the ends of these neurons. And with experience and maturation, there's a retraction or pruning of these connections, and therefore the concentration of these cannabinoid receptors drops down. So it is while this exuberant projection is being modified by experience and the environment that the system is most vulnerable. And that is one of the main reasons that the adolescent period is so vulnerable to marijuana effects. Let's see if I can get this going here. Looks like it has stopped. Oh, okay. There's another more recent publication looking at specific cortical areas. And this is mRNA expression of the CB1 receptor. And you can see here for P25, 40, and 70 across from rostral to caudal in the rodent brain. If we just look here at the difference of P25 to P70s, that is what the biggest change occurs, which areas show the biggest changes across adolescence. And you can see here, this medial prefrontal area has in fact 100% change in mRNA receptor density. Those more lateral areas of the cortex also undergo changes, but not nearly as robustly as that prefrontal area. And we'll see that in virtually every area of function, that prefrontal cortex is important. This came from a very nice recent review looking at cannabinoid levels and CB1 expression in infancy, adolescence, and in adulthood. And you can see here's the CB1, which we already talked about, expands during adolescence and then retracts down to an intermediate level in adulthood. This is across the whole brain. 2-AG, one of the endocannabinoids, is fairly high in infancy. It drops way down in adolescence and then returns to an intermediate level in adulthood. And AEA, or anandamide, which is another well-characterized endocannabinoid, stays fairly consistent throughout all of these periods. So you can see here the cannabinoid system is undergoing very robust changes during adolescence. Now we've talked, Scott also talked about the dopamine relationship, and it's shown here that in prefrontal cortex, the D1 and D2 receptors are very high throughout adolescence and drop down, while in the nucleus accumbens, they undergo gradual increase in infancy. They're peaking during adolescence and then drop down. So again, this relationship between the endocannabinoid system and the dopamine system undergoing rapid changes during adolescence, and that is the basis for the vulnerability, the selective vulnerability in adolescence. Just to look at individual areas now of the brain, the nucleus accumbens, caudate, putamen, and prefrontal cortex for anandamide and 2-AG. And from early adolescence, mid to late adolescence, you can see that anandamide increases. And in this study, chronic THC was administered at a very low dose every third day. And in the nucleus accumbens, there was an increase in anandamide concentrations in the animals that had received the chronic THC treatment. This did not occur in the caudate or in the prefrontal cortex. And you see here in prefrontal cortex, there's a gradual increase in anandamide content in this tissue. 2-AG has a totally different pattern of changes. And again, the acute or chronic, THC treatment did not affect 2-AG levels. That does not mean that there were no effects in the system, but just the levels were not influenced. Now, another recent review looked at the interactions of the possible ways in which the cannabinoid system could influence neural activity. And this relates very closely to what Scott already talked about. And in prefrontal cortex, we have a GABA interneuron. Now, GABA is inhibitory, and its normal job is to inhibit the glutamate neuron down here in pink. If you add THC to the mix, the CB1 receptor will then activate voltage-gated channels and stop the release of GABA, stop the inhibition produced by GABA. And that will result in a hyper-excitable glutamate neuron. The postsynaptic neuron is hyper-excitable. And this then translates downstream in the ventral tegmental area, for example, an increase in glutamate activity, glutamatergic neuron activity, then releases more glutamate into your dopamine neuron over here, and producing a whole host of second messenger systems and so forth. Also, there's a production of this of 2-AG from that postsynaptic site, which then feeds back onto the GABA neuron that is also there, inhibiting the inhibitory neuron, thus setting up a dysregulation of the glutamate GABA tone. Okay, there's a dysregulation of the homeostasis between glutamate and GABA, resulting in a hyper-excitable dopamine state. Excuse me. This slide represents some of the neurochemical changes which have been characterized from adolescent THC exposure in the prefrontal cortex. We talked about changes in GABA levels, increased pyramidal neuron fire, a whole host of other changes. I want to just emphasize here a reduction in LTD. Now, LTD, long-term depression, is responsible for intimately involved in pruning of synapses, and so there's a dysregulation there. Also, there are alterations in oscillation patterns, disruption of gene network associated with morphogenesis, cytoskeletal organization, and dendritic development, as well as a premature pruning of dendritic spines. All of these things occur in prefrontal cortex secondary to THC exposure in adolescence. This increase in firing rate then, of course, translates into a hyper-dopaminergic state in the ventral tegmental area, as well as in the nucleus accumbens. That's your reward center over here. This slide also talks about changes in the hippocampus, very different from that in the prefrontal cortex. Decreases in cannabinoid receptors, changes in serotonin system, and also here I'll point out the decrease in LTP. Again, LTP is important. Long-term potentiation is very important in the encoding of memories and in regulation of synaptic plasticity. So animal studies have shown that this synchronized neural network activity in the cortex develops during adolescence, and it's necessary for normal cognitive and sensory processes. And there have been a few studies now that show that early adolescent cannabis exposures permanently alter cortical oscillations with the selective sensitivity of the prefrontal cortex. Also, oscillations in the hippocampus are altered. The endocannabinoids control hippocampal networks. And in the normal animal, the hippocampus displays a theta rhythm, which regulates, I can't see my screen down here. Okay. Which regulates the nucleus accumbens as well as the VTA. And together these produce what are called sharp wave ripples. And these stabilize recently learned behaviors. Chronic THC or cannabinoid administration alters these sharp wave ripples because the theta rhythms are impaired in the hippocampus, and this whole circuit is dysregulated. So synchronized oscillations between cortical and hippocampal regions are required for information processing and behavioral tasks. And this stabilization of recently learned behaviors is one of the main effects that we are finding reported as a result of adolescent THC exposure. Now I'm going to talk about the prefrontal cortex development in adolescence. I'm sure you've all seen this figure out of Jay Geed from the NIMH. Beautiful, showing a cortical thickness from age 5 to age 20. Red and yellow are thick cortices, blue and green are thin. And as you can see in the human being across adolescence, the cortex gets thinner and thinner, with the last area to develop being that of the prefrontal cortex. Now, what's going on in the prefrontal cortex that it's getting thinner and thinner? Well, first of all, early on in childhood, the primary somatosensory cortex develops and the motor cortex develops. The motor system is very easy to understand. As the child moves around, there's that experience dependent plasticity that occurs in the motor cortex. So the efficiency of that system increases with use. If you immobilize an individual, you will have an abnormal development of that whole entire motor system and will never recover. Well, the same sort of thing occurs in the prefrontal cortex, but instead of motor and sensory, we have cognitive stimuli, social stimuli, and emotional stimuli. That these stimuli enter the prefrontal cortex, and those activated then undergo experience-dependent plasticity and improve their efficiency and stabilize those connections. All of this plasticity is due to both the influences of the glutamatergic, excitatory glutamatergic, and inhibitory GABAergic influences on spines from pyramidal neurons. Now, this is not exactly correct. The GABAergic influence is on the stem rather than on the spine itself. But the point being that both the glutamatergic and the GABAergic terminals are influenced and controlled by the CB1 receptors and the endocannabinoids, so that when you interfere with this whole process by adding THC into the system, you disrupt the normal maturation of the prefrontal cortex areas and the circuits that they are involved in. So I, being an imager by training, I always had to look at the structural and functional neuroimaging studies. And there was an incredible review published in 21 of 90 studies and over 9,000 participants. Review of all the neuroimaging studies that have been published with adolescent THC or adolescent marijuana. And they found that the effects, depending on the age of onset, the potency, and the duration of abstinence prior to the scan. So this is a big problem, of course, with marijuana, is that if people are smoking up just before they go in for their test, their cognitive test or their scan, it's going to influence the results. But basically, this review found very good evidence for functional and structural changes in 3 areas that relate to the prefrontal cortex: frontal parietal, frontal limbic, and frontal striatal. So these 3 main networks across all of these studies, 90 studies, show fairly consistent effects of adolescent marijuana smoking. Also, of course, the cerebellum. Now I'd like to talk about the effects of cannabinoids on cognitive function in adolescence. There have been several studies. There was a few years ago a meta-analysis of over 6,000 individuals that were grouped into those that started smoking marijuana before age 15, between 15 and 17 years, or after 18 years of age. The achievements were categorized, and the results show a significant interaction between age of onset and all of the outcomes. Those individuals starting smoking prior to smoking marijuana prior to 15 years of age showed the poorest achievement, and those after 18 showed the best achievement. Also, increasing frequency of use was significantly associated with the lowest academic achievement. Therefore, early cannabis use leads to poor academic achievement. Then there have been many, many other meta-analyses of this topic. They're shown here, 13 studies, 30 studies. This last one was an analysis of the meta-analyses, and they found that the largest effects of cannabis were decrements in learning new information. And bing, this relates back to that little cartoon I showed you in the hippocampus, where THC interferes with the formation of new memories. However, as interesting as this is, these are all cross-sectional studies, and it's really difficult you cannot say cause and effect, anything about cause and effect from cross-sectional studies. So the prospective studies are really what we need to look at. Now, in 2012, there was a very large prospective study published, the Dunedin study, which was really underpowered and came into a lot of criticism. And since then, last year, actually, there was a really nice study out of University of Minnesota, which prospectively looked at 86 participants ranging in age from 9 to 23 years at baseline. So they were primarily young. It was a 10-year study. The participants were interviewed in 2-year intervals, and they collected pre versus post IQ tests and so forth. And they also looked at alcohol and nicotine effects, and they controlled for parental education and so forth. And the bottom line was they found that there's decreased verbal learning and memory in the individuals once they start smoking marijuana. Now, what about animal effects? There have been a whole host of studies that I'll talk about, novel object recognition, spatial, lots and lots of effects, basically related to impaired plasticity. There was no impairment, though, in some other aspects, the Morse water maze or other aspects. I just want to highlight 2 studies, mostly because they compared directly adolescent and adults. This one is an old study where they administered THC for 3 to 6 months, and then there was a 1 to 3 month period of abstinence. And there were learning impairments in the young rats, but not in the adults. And this other study, where the animals received a 21-day treatment in either adolescents or adult females, and then underwent 21 days of withdrawal. And they found poor working memory in the object recognition test in adolescents, but not in the adults. And of course, this group has done a lot of this work, and it's really beautiful work that you guys have done. So anyway, there's been several reviews, not actually recently, they're older reviews, where they looked at a whole host of animal studies. This is novel object recognition, and you can see that some show memory impairment. They've looked at place recognition, Morse water maze, and so forth. All of these, there's various results, but the bottom line is that animal studies show that cannabis exposure in adolescents can produce long-term deficits in cognitive function. So now I want to talk about mental health in adolescents. And first of all, we'll talk about depression. There was a review last year published on 280,000 youths were surveyed, and they found that there was an increased association of suicide ideation, plans and attempts at suicide with increasing marijuana use. There were mixed associations in other studies. Generally, females are at greater risk of suicidal thoughts. And what about anxiety? Well, there's a scoping review of 47 studies published in 2020 where they only took studies that published frequency of marijuana use and dose of marijuana use, and where most of the subjects were under 25 years of age. And in this review of the 47 studies, half showed a positive association between cannabis use and anxiety, meaning the more cannabis used, the greater the anxiety, while the other half showed either no relationship or a negative association. So there's quite a lot of debate now as to the association between cannabis use and anxiety. Also, adolescent exposure has been reported to cause mood disorders. Females using more than once a week have shown psychological distress and more suicidal thoughts than the male counterparts. Other reports of conduct disorder, aggression, and antisocial behavior have also recently been published. With all of these, it's impossible to demonstrate cause and effect without prospective studies, except in animals. Of course, we can demonstrate cause and effect in animals. And here are, again, are all of these animal studies that are listed in adolescent THC or other CB agonist using elevated plus maze. This is open field, and you can see either no significant effects or increase and decrease in anxiety. Other tests, the forced swim test, which is related to depression phenotype, sucrose preference, as a sucrose preference. Again, the results are up and down and no effect. So while animal studies are great for looking at cause and effect, the results are not very clear cut. So we've been looking for several years at the effects of early stress on the endocannabinoid system. And this is one paper that we published a few years ago, looking at the frontal cortex or the hippocampus in animals that did not receive any drugs at all, but they were sacrificed on approximately mid-adolescence. And we have here in the green bars, males that were born in our laboratory from unstressed moms. The moms had arrived in the laboratory many weeks prior to insemination. And these are vendor derived. So these are animals that were born actually in Raleigh and shipped up to New York. And we don't know what their prenatal stress history is, but they certainly were stressed on P21 when they were shipped. And in these animals that were otherwise treated identically and housed in our vivarium, we see there's a huge reduction in CB1 receptors in frontal cortex in the males. And in those female litter mates, we found an increase in CB1 concentrations. We were surprised. In a hippocampus, this shipping had no effect on hippocampal CB1 receptors in males, but you see here it did produce a significant increase in females. So once again, we see big sex differences in the basal endocannabinoid system based just on stress history of the animals. We further went on and we published last year one of my pandemic papers where we looked at this whole issue in detail. And I'm not going to focus on this except for just to pick out these two studies, the Viveros and the Rubino groups. Here, there was no prenatal stress. The animals were born in the laboratory of moms that had been in the laboratory and not shipped. The pups underwent maternal separation on P9, and they were assayed in mid-adolescence. And you see all of these sex-specific changes in CB1 receptor in the males and in the females, increase in frontal cortex, amygdala, these are mRNAs, and then decrease in the females. But if this other group, they typically ship their pregnant animals, and they use the same identical maternal separation paradigm for the pups. This group looked at the adults, but here they found no changes in CB1 receptors in either males or females. So it could be that what we're looking at is this early stressor blocks the effect of that maternal separation stress on the on the neonate to produce no change in CB1 receptors. Obviously, there are things going on here, quite a lot of things going on. But when we're sitting here looking at and trying to understand and make sense of a lot of these papers, we need to look at the early stress histories of the animals in order to really identify what's going on. Now I want to talk about psychosis. This is a big problem in terms of effects of marijuana on mental health. And actually this year, there was a meta-analysis that published 6 longitudinal studies, an analysis of 6 longitudinal studies, where they found the increasing frequency of cannabis increases the incidence of schizophrenia. Daily users showed a 3.2-fold increase in schizophrenia, while daily users of high potency products increased the risk to 4.8. So this is a big increase with increasing potency of THC. Other groups have looked at age of first episode psychosis, and this is a paper that was published a few years ago by the UK group, and they've done a lot of work in this area. This bar represents those who never used cannabis. In this study, it was age of 31. Here we have individuals who smoked hash at weekends or hash every day. And these are individuals that smoked skunk, which is a very potent extract of cannabis at weekends, or skunk every day. And you can see here, those who smoked high potency marijuana products daily show an advanced age of first episode psychosis of a full 6 years. This is a very significant finding. This group also has gone through and looked at the incidence of schizophrenia in 11 sites across Europe and Brazil, and the contribution of frequency and potency of the cannabis used. So down here, this is the rate or odds ratio of someone having psychosis in an individual who never smoked. This is rare use individuals. These are individuals that use about once a week. And these are individuals that use cannabis daily. These are the data from marijuana with THC content less than 10%. And these are marijuana THC content greater than 10%. And you can see here that the daily users of the very high concentration THC show a huge increase in odds ratio of psychosis. So this is published in 2019. Animal studies have shown administration of WIN early in adolescence or in adulthood, and testing for pre-pulse inhibition, which is sensory motor gating, which is a behavioral manifestation of psychosis. This occurred in adolescents only. The adults did not show this effect. And in 2020, Dunn et al. Reviewed animal models of schizophrenia. They went through about 6 different animal models, a beautiful review, and showed the interactions with cannabis exposure and how cannabis, for the various analogs, increased the intensity of the psychotic-like behaviors. Also, they showed protection with CBD. There was little consensus about the mechanisms. But I just want to remind you all of this cartoon with the hyperdopaminergic state. This certainly is the underpinning of the increase in psychosis, because of course, you know the importance of the dopamine system in psychosis. And lastly, I just have a couple of slides on addiction. Cannabinoids, of course, are addictive. Cross-sectional and longitudinal studies show early use increases addictive disorders. Adolescents are more vulnerable than adults. 17% of adolescent daily users develop cannabis use disorder, while only 9% of adults do. Adults who used before age of 15 were 6-fold more likely to be dependent on other illicit drugs. And studies have shown that reward processing is abnormal. Chronic use leads to increase in anhedonia and apathy in adolescents, but not adults. Animal studies also have shown a variety of agonists administered increase self-administration of all of these drugs. And it looks like THC reprograms the nucleus accumbens transcriptome, affecting reward processing, impulsivity, and aspects of cocaine taking behavior. And in conclusion, marijuana interferes with the normal brain development, cognitive function, mental disorders, especially psychosis, drug dependence, and future drug use. There's an interference with cortical and hippocampal oscillations and a maintenance of this hyperdopaminergic state. These are the underpinnings of the neurobehavioral disruptions that are described. Plasticity in the hippocampus, which is the underpinnings of memory, remains sensitive to marijuana throughout the lifespan. And all of these phenomena are related to the amount of marijuana that is consumed, the amount of THC that's consumed. And I just want to thank my funding sources, and I'm sorry I ran over a little bit.
Well, thank you, Diana. That was marvelous. Very comprehensive look at the risks and mechanisms of cannabis and THC effects during adolescence. So now we're going to take another look at adolescent risk. And Dr. Nicole Schramm-Sapyta will be leading a youth panel discussion. Dr. Schramm-Sapyta is Associate Director of the Duke Institute on Brain Sciences and will be moderating the panel. Nicole.
And before I start, Wanda's going to give a little preface for me.
Yes. So with the discussions that we've had so far, especially the scientific and research information that we've been given, I think that there are a couple of things that I'd like to interject before we go on. The question might be, well, if we have known this, what are we doing about it? And what does prevention look like? And is anyone thinking or talking about that? So I won't have an opportunity to talk about this a little bit later on, except if you want to stay for the discussion. But I just wanted to point out a couple of things to you. One is that the Durham, County Department of Public Health, every 2 years issues the Youth Risk Behavior Survey. It's the document that TRI uses in order to develop strategies to prevent all kinds of things. We're talking about marijuana today. So just to let you know, in the most recent document, which can be found at healthydurham.org, middle school students, and this is over 1,000 of them, indicate that 10% of middle school students have used marijuana in the past 30 days since the survey was conducted, and 26% of high school students. I want to say that the number for middle school students have gone down, and for high school students gone up from 22% in the last survey to 26%. So Together for Resilient Youth has a youth coalition and a college age coalition. The youth coalition, ages 9 to 17, become peer-to-peer health ambassadors to speak about the dangers of substance use, including marijuana. And we also look at Search Institute, Search Institute developmental assets. Really important, because Search Institute developmental assets, the premise behind that research is that when young people have these assets, then regardless of socioeconomic status, they can avoid some of the dangers and pitfalls that we see. So I'll be talking more a little bit later about that. But this is what I wanted to say and provide this information if you would like it. It's a one-page document from Colorado on marijuana use, which we have used since 2015. And if you want to know a little bit more about the specifics about use and what that looks like in the real world, this document indicates regular marijuana use by adolescents and young adults, as we've heard, is associated with schizophrenia and bipolar, which they've published in Colorado in their public health statements. But this is something that I think will resonate with all of us. Heavy marijuana use is defined as daily or near daily use, 5 to 7 days a week. Regular marijuana use is defined as weekly, 1 to 4 days a week. Occasional marijuana use is less than weekly. Why is that important? It's because regular marijuana use is associated with many of the things that we just heard. And so what we want to do in terms of prevention is to make sure that youth have the skills and the information that they need to make the choices that they can make. If youth continue to believe that marijuana is good because it's green and from the earth and it's like grandma's marijuana, well, that's a misnomer. And I would also like to petition for this, not lobby, petition for this. And that is, we were able to reach over 900 students through driver's ed courses to provide this information specifically about how much marijuana, because our young people, 26% of them that admit to it, are using marijuana. So I just wanted to bring those up before we enter into this terrific, wonderful, that I've been waiting for panel with Nicole. So Nicole, thank you for giving me the opportunity to say those few things, and please go ahead.
All right, thank you so much, Wanda. And I want to invite Connor, Olivia, John, and Brianna to turn on their cameras now. It's your time to shine. So I want to say that we are so honored this morning to have 4 college students from Durham here to speak with us. So they are hopefully, well, they're all definitely in the late adolescent phase, and they have recently completed the early and mid-adolescent phase. I'm going to ask each of them to just go around at this point and just tell us your name, what college you go to, what year you are, and what you're majoring in. So let's start with Brianna.
Hi everyone, my name is Brianna. I am a Duke student majoring in neuroscience, and I'm a sophomore.
All right, Connor.
Good morning. I am a senior public health education student, and I go to North Carolina Central University.
All right, Olivia.
Hello, everyone. I am Olivia Harris, a senior public health education major at the historic North Carolina Central University.
And John.
Hello, everybody. I'm John. I'm a senior graduating this year, and I'm majoring in neuroscience and computer science at Duke University.
All right, so lots of people who should know all these things. First of all, I just want to get each of you maybe a little bit from Duke and a little bit from NC Central, because those are good representatives of Durham. What is the culture like on your campuses? Is drinking the norm? Is marijuana usage the norm? Just what's available and how often do people do it? Olivia, I'll let you start.
Yeah, so outside of it, being common, describing the drug and alcohol culture, it's frequently used. In a way, we use it to fellowship with one another. It's a way for everyone who may not have a common interest when engaging in conversation or activities with one another to share a drink as a way to celebrate a moment. Whether it's like a grand activity or just something small.
Connor, go ahead. What would you add about the culture at Central? And then we'll come to Duke.
Like Olivia said, I know a lot of people use alcohol and marijuana as a way, like set up fellowship. I know they use it in a way of celebration, and it is typically the norm. You'll see or you might smell marijuana throughout campus, in the dorms and things like that. And they substitute it with vaping. when they're going to class or in a building where marijuana is not allowed. So it's definitely widely used on campus.
Mm-hmm. All right, John, how about the culture on Duke's campus?
Yeah, in terms of alcohol, I think like many college campuses, use is pretty widespread, whether in social functions or before a concert or pre-gaming for Duke basketball games. But with regards to marijuana, I've lots of peers and friends that use marijuana regularly and frequently, and I have many more who don't. So I would say use is by no means ubiquitous, but it's not hard to find, and it is widespread. There is a culture on Duke's campus.
Bree, would you add anything to the Duke description?
Yeah, I would definitely echo what has already been said. I think my understanding is that most substances are available through some indirect source, but not all substances are equally prevalent. So I agree with John that alcohol is like definitely widely consumed across campus and seems to be purchased underage through like either upperclassmen or through the use of fake IDs. And then of course, marijuana is a different story since it's not legalized in North Carolina, but it seems like people can still definitely get their hands on it if that's what they choose. But there are just more hoops to jump through, I guess. And I've also noticed that since it's not legalized, it sometimes leads to the usage of other synthetic marijuana, which poses many threats. And then in terms of other like hardcore drugs, quote unquote, they seem to be used far less frequently. But yeah, that's all I would add.
So it sounds like we can summarize to say everything's available and there's a range of usage from only for celebrations to daily use. Probably some people that never use any of these substances on both campuses, I would imagine. In terms of the messaging from your university leadership, the student health or the campus rules, how are things communicated from university leadership? We'll just go in the opposite direction. Bree, I'll let you start with that.
I never recall discussing preventative approaches towards like marijuana or alcohol usage on campus. I remember taking like a very short 2-hour-long course as a freshman introducing our alcohol policies. But since that point, I don't recall any communication with administration on that topic.
Mm-hmm. John, what about you?
Yeah, I'm involved in Greek life at Duke, and I know that the administration has been targeting Greek life as a sort of intervention for binge drinking and alcohol education. So I have had a lot of more extended courses than just that mandatory 2-hour freshman seminar what Bree was talking about. And throughout those courses, I think the saying that they have is ride the wave. So encouraging and recognizing that college students will drink, but encouraging responsible habits and not binge drinking. In terms of marijuana, I think those trainings don't really address anything about marijuana. And I have never heard any educational materials from the administration.
All right. What about at Central? Connor, would you like to start?
So at Central, like you said, I know we definitely have classes and courses regarding alcohol use, and being a public health major, we definitely speak on marijuana and drug usage often. So our teachers definitely, bring that to our attention and, get our opinion on it and let us know that it is a problem on campus. As far as administration, I know that they're strict when it comes to usage in dorms. I've been in dorms where we've had police walk through the building with dogs, sniffing out, and that I've seen people get arrested for it. I've seen people, definitely get in some serious trouble about it. So they don't take it lightly. And, one of the people that I know got arrested, he had to do community service for a while to even be allowed back on campus. So it's definitely, even if it's not spoken on as much by the administration, they definitely enforce those rules against using it.
All right, Olivia, what about you? What have you observed?
Just to kind of piggyback off of what Connor said, it's heavily enforced at our institution. I know like during mass events where alcohol is allowed, they do ask that, you don't drink and drive. We have like a few posters and like on a big screen in the gymnasium. It has like, don't consume like a lot of alcohol and things of that sort. While so personally, I've had to take many classes. So I'm a resident assistant here at my university. So like we have like during our trainings, like maybe several classes where we have to be properly trained on how to deal with situations where, individual students who may get caught using drugs and how to report, like proper protocol and stuff like that. So
in your role as a resident advisor, sounds like you've learned a lot. And then how do you communicate that to your residents?
So we have monthly like hall meetings that pretty much like gives them an overview of what's going on, the use of marijuana, because like Connor has said, we smell it frequently in our halls, and that kind of brings attention to like our administrators. They may just walk in the dorms and they, say, hey, this is an issue. So now we have to, perform room checks and things like that. So when we have to do that, and I'm talking about, it happens really quickly, like when our vice chancellor comes into the building and they say, this is what we have to do. And, 5-6 students might be smoking in their, bathroom. The police gets called and now, they have to get written up, like a accident report has to be documented, and they get penalized for it. Typically, they will lose their housing. And like Connor said, in order to even be allowed back on campus, they have to complete, I want to say, 65 community service hours. So.
Wow, that's intense.
Yeah.
So much more of a enforcement approach. Do we see similar approaches at Duke, John or Bree?
Yeah, I just wanted to step in because I'm very surprised to hear this. From my understanding at Duke, they're pretty lenient with their disciplinary policies. So if people do get caught, yes, the Durham police could be brought in, or the Duke police could be brought in to handle the situation. But almost always it ends up being a slap on the wrist from the Duke student conduct. So I don't think I've heard of anybody being arrested on campus or anything like that.
Wow. Yeah.
Oh,
wow.
I think the only thing I would add is I have heard of people being arrested or disciplined for the usage of their fake IDs, but not for consuming it to start with, if that makes any sense.
Mm-hmm. Well, there's a lot to dig into there. Do you think students are aware of the dangers? So we just saw the great talk from Dr. Dow-Edwards. It could increase depression. It can inhibit learning. We're going to learn a little bit later, it can have effects on unborn children, even before they're conceived. Do you think people are aware? And I'll actually start that conversation with our public health specialists. So are people aware of these dangers? The research is pretty new, but is it out there?
They are. I guess I'll start since the order. They definitely are aware of like the health risks of marijuana. However, they also focus more on like the benefits that come with marijuana. So like I think we all heard like it reduces like anxiety and like relieves pain, and of course like it is a treatment for cancer. So they kind of use those facts and they kind of run with it, not looking at like the negative attributes that come with, of course, consuming marijuana. So. Mm-hmm. Yeah,
that's definitely true. I think another thing is our generation is very much a show me generation. So while, they might have classes, they might learn things like we've learned today with, all the brain functionings and things that it can affect. I think they have to see people, like personally see people being, mentally affected and things by marijuana in order for it to really, click with them and be like, okay, maybe I need to regulate how much I use or stop altogether. But they definitely know the risk. But like Olivia said, they choose to look on the brighter side. And as long as nothing's physically wrong with them, they think they're okay.
Mm-hmm. So it's not me, therefore it doesn't matter.
Pretty much, yeah.
Mm-hmm.
Right.
Well, I'll turn to our two neuroscience majors now. It's in many ways, it's hard to talk about the brain science because it's so complicated. How do we take that message to students like you guys, like all of you and your peers who are experimenting and not feeling negative effects yet? They may not appear until years later.
I think Connor hit the nail on the head in terms of like needing to see the outcomes, these negative consequences, because even within the neuroscience department and within my major, my peers within my major, we all learn about this science intricately and we know it, but it's just like the blissful ignorance of youth. You don't really realize what it means. So I think that's a problem to continue to think about, is how you can reach undergraduate students and teenagers, high school students.
Takes me back to Crystal's talk this morning. Maybe we need an ethnodrama. Go ahead, Brianna.
I was just going to say I agree with everything that's been said already. And I think that part of it is just talking about it and spreading awareness rather than shoving it under the rug is not a big issue. Because what I've understood based on my conversations with peers is that surprisingly, people who come from states where marijuana is legalized seem to be the most knowledgeable about what they're consuming and the potential risks, because they just like have learned to understand what they're consuming. And so I think like that sheds light on how important it is to actually have those conversations and recognize all aspects of marijuana consumption.
That is the perfect lead into the final question that I want to ask all of you, which is, one of the things that spurred this whole symposium was marijuana is getting legalized in more and more states around the country. So if North Carolina were to legalize it, would that change things on your campus? And if so, how? Connor, I'll let you start.
As far as changing things on the campus, I feel like people just be more comfortable smoking, and it would definitely, I feel like it would increase the amount of people that smoke. I feel like it would also be safer if it was legalized, because as I spoke before, there are a lot of, back doors and back end ways of getting marijuana. So I just feel like while it might increase for a little while, I feel like it would definitely be safer for the students. So it's kind of, whichever way you want to look at it, it's good and bad. So,
Olivia?
People of color are already in prison at, disproportionate rates for nonviolent crimes. So legalizing marijuana would decrease the jail population and reduce, disproportionate incarceration of minorities. As we stated before, I've seen alone this year about, give or take, maybe like 35 arrests, just within like a month. We're not even fully in our semester. And so, yeah. And then like Connor said, it would definitely be safer and people feel, a lot more comfortable smoking. They don't have to be sneaky about it or feel unsafe.
Mm-hmm. So a lot less of that police intervention that is probably pretty disruptive to residents' life.
Yeah.
And one more thing, I just feel like some people do it because of that sneaky aspect. They feel like they're in college, they're free, they're away from their parents. They want to do a lot of things that they can't do at home. So I feel like legalization might help, discourage some of that just sneaky behavior and wanting to do it just because you're away from your parents.
Mm-hmm. Takes away at least one incentive to do it. Mm-hmm. John and Bree, thoughts of how it would change things on Duke's campus?
Yeah, I think it wouldn't change too much, to be honest. And it's crazy because, again, if we go back to our understanding about the potential risks and also talking about the response that I've seen from Duke student conduct, nothing really happens unless you're a repeat offender over and over again. On the other side, so if you're smoking weed on campus inside the East Campus loop and get caught as a Duke student, it's a slap on the wrist. But if you're outside of that wall and you're a Durhamite and you're not a student, then you can face criminal charges. And I think that kind of double standard makes no real sense. What do you think?
Yeah, I agree. And it was really shocking to hear everything about NCCU's culture. Thank you for sharing. I agree with what John has just said. I don't anticipate there being a huge uptick in the number of people who start to use marijuana if it were to be legalized. I think there might be an increased number of like first-time users, but I don't anticipate like regularity after that point. And so I think it's really just about creating a safer environment for it to take place, because as we were talking about earlier, people who do want to smoke are finding ways to smoke. And so that population would continue along that same trajectory. And so I think it's honestly just about keeping everyone safe, making sure that the substances that they are consuming aren't laced with other harmful substances as well.
All right. Any final thoughts before we hand it off to the next session? You guys have been absolutely wonderful. Thank you so much. We've been so honored to hear these firsthand experiences, and just from people who are on the path to becoming wonderful educators, informed citizens. We're so excited that all of you are doing well in college and focusing your efforts on such important questions for our society. So I want to say thanks to Bree and John and Connor and Olivia. I really appreciate y'all taking the time with us this morning, and just thank you for being so candid with your insights. I really appreciate it. And I will hand off now to, I believe it's Ed for the next one.
Thanks, Nicole, and thanks for leading this, and thanks for all the participants in the panel. And it's great to hear from the next generation. And speaking of future generations, now we're going to look at how marijuana affects families and cross-generational effects. And so, we heard a bit earlier from Scott's work and a bit from others about effects of marijuana and THC on early development, and now there's increasing evidence even before conception that there are effects not just if the mother smokes, but if the father smokes as well. And we're going to hear now from Dr. Susan Murphy, who's associate professor in obstetrics and gynecology, and she's a director of the Cyphers Center funded by the John Templeton Foundation. So that's a center here at Duke having to do with assessing the risks of does what dad do matter too? So is it are there effects of marijuana smoking on sperm quality and imprinting, and is that conveyed to the next generation? So Susan?
Thank you, Ed. Just getting the screen up here. Can you hear me okay?
Yeah, that's fine.
Great. It's truly a pleasure to be here today. I've learned a lot this morning, and I really appreciate the students' discussion. That was very insightful and helpful as we've been thinking about trying to kind of inform the public about our findings. It's great to hear from the young people what's on their minds in terms of marijuana legalization and usage. So I'll be talking today about work that we've been doing in the Cyphers Project, as Ed mentioned, on the generational effects of cannabis. So just an overview of what I'm planning to cover. Pretty simple question. Why do we want to study cannabis use in males? Does cannabis use change sperm methylation? I'm going to talk about methylation, hopefully explain it in a simple way that makes sense. Are changes transmitted to offspring, and are the changes that are in sperm, are they permanent? So first off, why should we study cannabis use in males? Well, there's several good reasons, I think, many of which have been covered this morning. So Number one, cannabis legalization is expanding. And with that has come an increased perception that cannabis use is safe with increased usage. So we now have 18 states and the District of Columbia that have legalized recreational use. We have 37 states that have legalized medicinal use, and it's been decriminalized in 13 states, including here in North Carolina. Another reason that has been discussed earlier is that cannabis potency is increasing. So in contrast to the days of the hippies, when the THC content in cannabis was less than 2%, nowadays it can be upwards of 28% or so. And that was from a study done in 2017 and from cannabis in dispensaries available in Colorado. Another good reason to study cannabis use in males is that males are the major users. This chart plots male and female use over time through 2016. The red line shows use in males, the green line is in females, and blue is the average. So we know that males are the dominant users. And the other part of this is that actually reproductive age males are the most common users. So males of reproductive age, meaning when they are producing sperm, which happens throughout the course of their life from adolescence onward, are using cannabis. And then I think the major reason is kind of summed up here in a piece that came from the Harvard Gazette in 2020, what we know and don't know about pot. And I love this quote, which definitely sums up my thoughts on it. There's been broad public experience with pot, but the medical community still doesn't know enough about the health effects. And what it does know is often obscured by enduring myths. So this is another reason in my mind, and one of the primary reasons that we need to study this, so that people can actually then make informed choices about when they use it and whether they use it. So my lab has been focused largely on something called the developmental origins of health and disease, which is a hypothesis that it revolves around the idea that the environment that we encounter during our early development can lead to changes in the way that the genes in our DNA are used, are regulated, and that this may be somatically heritable, meaning that as cells divide, those changes are actually transmitted from one cell to the next and to the next. And that can increase the risk of later life adverse outcomes. And this has been kind of caught on fire, this whole idea. And there are thousands and thousands of studies that have been published looking at the in utero environment and how that impacts later risk of disease for the child. So the primary focus has been really on pregnancy and mother's exposures, which makes sense in terms of the hypothesis. But my group is really interested in what the dad might be contributing, and are the dads contributing anything? And this became kind of a question that started emerging from anecdotal evidence that auto mechanics children who have a high exposure to the hydrocarbons have a higher risk of developing cancer. Other studies have shown that advanced paternal age is associated with an increased risk of miscarriage and of having a child with autism, as is paternal obesity associated with the increased risk of having a child with autism. So what is it that dads could be contributing that might increase these risks? So in fact, what is it the dads might be contributing or might be exposed to even before he has children that could impact the health of his children? And how might this be transmitted? Of course, dad contributes a sperm to the child, to the development of the child. And within the sperm is the DNA sequence. DNA sequence is obviously very important in controlling development. We know that when fertilization occurs, that one fertilized cell is considered a stem cell, and it will eventually give rise to the formation of over 200 different cell types that have unique shapes, unique functions throughout the course of the individual's lifespan. But it's the same DNA sequence that does this. So there must be something else aside from the DNA sequence that is causing the cells to differentiate and behave in the way they do to form all the different 200 types that are in the body. And what what we think is happening, or what we know is happening in the majority of these cases, is epigenetics. So epigenetics is a form of gene regulation that doesn't involve the gene sequence, but does impact how the DNA sequence is used. So in sperm, this is just a complicated figure to show sperm, the DNA that's packaged in the sperm, and the proteins and modifications associated with it. We refer to these modifications as the sperm epigenome. And these are just the chemical tags that carry the instructions for how the DNA should be used in development. And these modifications are actually really important because there's about 3 feet of DNA, if you stretched it end to end, that have to be packaged into the head of one sperm cell. That's a lot of condensation that has to happen in order to get the DNA to fit. And these epigenetic modifications, these chemical tags, help to condense the DNA to make that work. But they also have another important function, and that is to regulate gene expression. And what I'm going to be talking about today is DNA methylation, which is shown here in this red oval. So what's DNA methylation? When I talk about that, what I mean is a methyl group, which is a carbon and 3 hydrogen atoms, and we depict it here as a red circle. These methyl groups are little chemical tags that get added to the DNA, specifically at regions that are that have CG dinucleotides. So what happens is the C in the CG sequence gets the addition of a methyl group, and because DNA is double-stranded, the top strand and the bottom strand are complementary to one another. So actually reading either strand, the DNA is methylated on both the top and the bottom, or the Watson and the Crick strand. So what does that look like in terms of a gene? So if this is just the DNA double helix, and this is a gene, and we have methylation. These methyl groups have been added to the body of the gene, but near the front end, the business end of the gene, where the DNA or where the cellular machinery assembles to start using that gene and to produce a product from that gene, there is no methylation. And when that happens, in that context, that gene is generally active. So in contrast to that, if we have less methylation in the gene body, but the business end of the gene is actually highly methylated, this leads to a repression of gene expression, and the gene is in a repressed or inactive state. And this is normal throughout the body. All of our genes are marked in some way or another with methylation tags. And in fact, if we didn't have methylation, if it was stripped from the whole genome, that would not be compatible with us surviving or even developing. So methylation is a necessary process, but it can be affected by many things in our environment, a number of which are shown here. And the end result of that is that methyl groups can be removed from the DNA or added to the DNA in places they shouldn't be by our exposure to these different types of environmental, either contaminants or physical things like obesity and our diet and stress and sleep. But what we're talking about today is drugs and how cannabis in particular affects the methylation within the DNA. So the primary question we set out to answer when we started this study was, does cannabis use changed sperm methylation? And in fact, bottom line is it does. These are results from our pilot study where we had recruited 12 users of cannabis and 12 non-using controls who graciously provided semen specimens that were analyzed at the Duke Fertility Center and then underwent sequencing to profile the methylation at about 5 million of the CPGs or CG sites throughout the genome. What's shown here is the blue on the left with this heat map. The blue means less methylation, yellow means more methylation. And what you see is that actually in the user group, there's a fairly substantial loss of methylation. And these are the most significantly affected genes. These are genes that had 10 or more CG sites that were showing a significant loss of methylation. The gene list is on the right. I don't intend for you to read that, but what I'm going to talk about is one of these genes called DLGAP2. So DLGAP2 is actually an important gene in neural function. It plays a role in the postsynaptic density. DLGAP2 stands for disc large associated protein 2, and this was particularly of interest to us because this gene had been implicated in autism spectrum disorders, as well as in schizophrenia. And these are a few of the publications that have highlighted DLGAP2 as playing a role. What we did was confirm our results from that genome sequencing that we did using a completely independent method. I'm not going to go into the details, but it's a method to get targeted regional analysis of specific genes that we were interested in. And this graph here, which shows methylation on the y-axis and the specific CG sites, of which 10 were analyzed here on the x-axis, it shows that the controls, the gray bars, have higher methylation than the exposed group in the black bars. And this was significant across all of the sites that we analyzed using this independent method. So our results were validated. Now, what was interesting, we wanted to see if there was functional relevance to this methylation. So in order to do this, we used a bank of human fetal brain tissues that we have in the lab to examine the methylation at this specific region that we saw was hypomethylated or had loss of methylation in the sperm, and how this related to expression of DLGAP2 in the brain. And looking at the entirety of the group, we see that there's an inverse correlation between methylation and expression, where higher levels of methylation are associated with lower levels of expression. Because autism is a neurodevelopmental disorder that is 4 times more prevalent in males than females, we also were interested in whether or not there were differences by sex. So in looking at this, the males still have an inverse correlation, although not quite significant. The females, however, seem to be driving this relationship with a very significant inverse correlation between methylation and expression. We don't know what this means yet in terms of autism. We don't know anything about the history of these particular tissues that were used. We don't know the cannabis smoking history of the fathers or the mothers from this group. This simply just says that the methylation that we see at the region of this gene does seem to play a role in the way that this gene is utilized. We're also interested in other neurodevelopmentally related genes that came up in our studies and tested another 7 genes for the effect of THC exposure using a rat model. And this is work that was done in our host lab, Ed11, or at least that the exposure of the rat, the THC was done there. We did the methylation work in my lab. And of these 7 genes that we tested, 5 of them are showing significant differences in methylation. And again, these are genes involved in neurodevelopment. Now, what was also really interesting to us was we had another group of rats that were exposed to nicotine. In these males, the sperm was harvested, and we looked at these same 7 genes in the sperm from those rats and saw changes that overlapped some of the significant genes from the THC exposure, but now we're talking about nicotine. And if you're paying really close attention, you might have noticed that actually, while the THC exposure, which is the green bars, shows a loss of methylation at these particular genes, the nicotine exposure actually causes an increase in methylation at these same CG sites of the same gene. What this tells us is that these genes are vulnerable to exposures, to different types of exposures, and they respond differently depending on what the exposure is. And as one of the questions earlier, whether or not Dr. Parnell had looked at cannabinoids and when he was talking about alcohol, and I think Ed asked him if he looked at nicotine as well. That's something we are very keen on doing, is looking at co-exposure, because you could assume maybe looking at this that it maybe would negate the effects of both exposures if they were exposed at the same time to both nicotine and THC. And that's not something I'm advocating, but we are very interested to see how co-exposure actually impacts methylation at these regions and at other regions of the genome. So next, I'll address our changes transmitted to the offspring. This is a challenging question to address. We do have a human cohort that we are in the process of recruiting to address this in humans, but we have done some studies in rats. We know that exposure in the father rats causes neurotransmitter deficits and neurobehavioral deficits in his children, or the rat pups born to that dad when he was bred to a drug-naive female. And these are 4 of the papers that came out from the CIPHER study showing those effects, which I'll refer you to if you want to find more information out on this. We also looked at methylation, and the first thing we looked at was DL-GAP2 because of the results we saw in the human studies that were done. We looked at 8 CG sites in DL-GAP2. The rat and human sequence are not completely homologous, so we identified a separate region of this gene in rats that showed differences in methylation that are significant in the sperm. That's shown here. In the offspring hippocampus, from these offspring were derived from these fathers who were exposed relative to control offspring. We see a significant difference at one of the CGs, but it wasn't at the same sites that were affected in the sperm. In the nucleus accumbens, however, we do see a significant loss of methylation at one of the CGs that was also similarly affected in the sperm of the exposed dad. So there's some evidence that there is transmission to the next generation of this change in methylation in sperm. We also looked at several other genes and saw evidence of transmission. This gene on the left is PXYLP1. It's involved in glycosaminoglycan synthesis. On the top is shown the sperm from the father, and the blue and green bars are the exposed animals. They were exposed at different times relative to the time of harvest of the sperm. The black bars show the controls, and in all cases, you can see there's a significant loss of methylation in the sperm at these particular CG sites. We also see that if you look below in the offspring from those fathers who were exposed themselves as sperm, as developing sperm in their fathers, but their sperm is now also showing the same changes in methylation, this loss of methylation. So they're actually poised to then transmit that change to the next generation. And we are currently in that process of studying the next generation, the F2 offspring. We also looked at GABA-BR2, which is a GABA-A receptor family subunit. There's loss of methylation in the sperm that we saw in the upper right, and we saw a milder loss of methylation present in the hippocampus of the offspring there. Another gene we looked at is MTSS1L, which is metastasis suppressor 1-like. It is implicated in neurodevelopmental disorders and is an important, plays an important role in cell membrane organization and GTPase activity. In sperm, we see loss of methylation in one of those exposure groups who were exposed early, but then had a withdrawal period during which they weren't exposed following that exposure. We also see it in the pups, in the hippocampus and the nucleus accumbens, these same changes. So there is evidence of heritability of these methylation changes in the offspring. Now, this MTSS1L gene is actually pretty interesting. We looked again at the methylation expression relationships in the offspring of the exposed fathers. And here, if you look at this on the left, this is the nucleus accumbens of the offspring. It's one of the significant sites of methylation. And from looking at this in the exposed, which are the blue triangles, and the controls, which are the black circles, looks like there's no real relationship there between methylation and expression. But when you divide this out by sex, it gets really interesting. So in the males, there is a positive correlation, an inverse correlation in the controls between methylation and expression, with higher methylation associated with lower expression. But that's reversed in the exposed group. In the females, it's just the opposite. So it's almost as if the exposure has reversed the normal relationship between methylation and expression in the males and females. How that pans out in terms of functional role for this gene, we don't know yet, but I think that this highlights that there are going to be very important sex differences in how dad's exposure actually plays out in his offspring. So are the changes permanent? In order to look at this question, we need to understand that human spermatogenesis is a cycle that starts with the differentiation of a sperm cell that's going to go on and mature from the spermatogonia. And that process takes about 74 days in humans. That's important because we recruited another cohort of cannabis users and non-users and asked them, the cannabis users, to stop using cannabis for a period of time that encompasses one spermatogenic cycle, so 77 days. They provided a semen specimen before and after, as did the control non-user group. and then underwent, we took the sperm and isolated the DNA and analyzed every CG site throughout the genome using another sequencing approach, and in this time for 30 million CG sites. This is a summation of the significant results for each of the CG sites, which are shown by the dots in this volcano plot. The dots that have labels on them, again, I don't mean for you to read, but those are significant after adjusting for all the 30 million multiple comparisons that are being performed in this analysis. So that significance level is a p-value of less than about 3 × 10⁻⁹. The other thing I want to point out from this figure is it looks like there are more that are that significant in the before group than the after group. There are fewer adjusted p-value significant sites in the after, which was encouraging to us. We were hoping that that period of abstinence from cannabis use might actually reverse some of the effects that we were seeing with cannabis use. This is another way to look at the data. This is showing the same CG sites on the left, and they're plotted by the difference from the control. So the ones that are below 0 on the y-axis are those that have lower methylation in the controls than the users. The ones above 0 have higher methylation in the controls than users. And then what you see is on the right that those sites actually trended toward a more normal level, toward what we see in the control groups, because they're now more clustered around the 0 level of methylation, or 0 difference in methylation. In order to show that that change wasn't just a function of normal changes in sperm methylation over time, we also compared within each group, looking at the changes in methylation at these particular CG sites in the before versus after samples. And this shows, if you look at the bottom violin plot versus the top on the right, that there's a much narrower distribution in the controls over time, so much less fluctuation in the methylation in those in the control group than there is in the user group. So this provided or supports the idea that these changes are actually due to a remediation effect from cannabis withdrawal. When we did bioinformatics analysis of the affected genes and the pathways and functions that they might be enriched for in the before group, these are the different significant pathways and functions that came out, including things associated with the nervous system, cerebral disorder, neurodevelopmental disorder, agenesis, organismal development, growth, cardiogenesis, et cetera. And we did this with the after specimens too, just comparing the users to the control group. There are still significant genes that come out in the after, but most of those that had a significant difference in the before, most of those CG sites are actually ameliorated in the after. And there are other things that are showing up, many of which are still involved in neural function. We don't know yet what this means. We don't know if this was because of the withdrawal and stress that these men underwent during the course of the withdrawal and the weekly drug testing they had to do, but it's something that we plan to look at in the future. So overall, I've shown, hopefully convinced you that reproductive age males who use cannabis do have altered methylation in their sperm relative to controls, and our rat data supports that. The DLGAP2 gene, which is an autism candidate gene, shows hypomethylation in sperm with cannabis use, and that we do have some evidence in our rat model for cross-generational transmission. We've shown that other genes involved in neurodevelopment are altered by both THC and nicotine independently, and some of these are in opposite directions, and that abstaining from cannabis use for the length of a spermatogenic cycle does mitigate some of the methylation changes in the sperm. And our rat data, which I didn't have time to present, is also consistent with the human data. So what are the implications for this? I think one of the big ones is preconception counseling. So having clinicians and others, community members, talking to men and women about the harm that can come from cannabis use prior to planning to have children, so that men can make an informed decision about whether or not they should use, and also knowing that if they stop using, that might actually improve the epigenetic health of their sperm. We also think that these methylation marks that we're seeing could be used for preconception screening. If a man is interested in knowing what the chances are of passing on some of these epigenetic changes to his child, we could survey the sperm and look to see what kinds of changes are present. And then it also has implications for newborn screening. If we know that there's some heritability from the father who uses cannabis to his child, it may end with the associations with neurodevelopmental disorders and autism. We could use the methylation markers as a screening tool to identify children much earlier in their development who may be at increased risk for developing such neurodevelopmental disorders, allowing for much earlier intervention, which we know has benefit. With that, I'll thank the members of my lab, particularly Rose Trott, who's now a postdoc at Johns Hopkins, and want to tip my hat to Ed Levin's lab. We couldn't do this work without all the studies they've done, as well as John Mitchell and Scott Collins, who led the human cohorts, and many others. So thank you for your attention.
Well, thank you, Susan. That was an excellent talk, and showing you that the dads are important too. And as a dad, I'm happy to hear that. Although it's a responsibility that we need to attend to. Yeah. So now I am going to hand it off to Nicole, I think, who's going to introduce the next speakers.
All right. So our next speakers are very familiar to all of us. So Dr. Wanda Boone, who you all met earlier, is our co-presenter and co-collaborator, and her colleague Eve Puffer are going to talk about the impact of ACEs and coping together and family cohesiveness on marijuana usage. Go ahead, Wanda.
It looks like there it is. Okay. All right, what a wonderful conversation and session we've had so far. And because we talked about males and females and families and everything associated, we wanted to bring you this information about the impact of adverse experiences and the work of Duke Global Health Institute, Dr. Eve Puffer, and the program Coping Together. So what a wonderful image to look at right before lunch. But the purpose of my sharing this with you is because, as we've talked about marijuana and marijuana usage and the impact of marijuana on our physical bodies, in terms of community, we have to understand that we are all different in some ways, and one size does not fit all, except for what I see in Coping Together, and also with the prevention efforts that we set forth as Together for Resilient Youth. I think cultural humility is extremely important, understanding that we have different tastes. And when we come to the table, we have to be respectful and mindful of the differences that exist among us, adverse experiences. So this image represents the compound challenges associated with adverse experiences. Many of us are aware of adverse childhood experiences. And my association with adverse childhood experiences began in 2013, 2014, when there really wasn't a lot of information, or rather wide discussion about it. So in terms of what happens above, we have climate change, environmental situations, pollution, hurricanes, floods, droughts, and even COVID-19 is identified as an adverse experience, possibly because it's been experienced globally, but the impact of COVID-19 on people of color have been essentially different than the experiences of white people. And so we see a challenge there. In terms of adverse childhood experiences, I'm going to talk about that a little later. And then adverse community experiences, well, let me back up, because adverse childhood experiences include things like neglect and household dysfunction that really launch us into a significant number of outcomes that are adverse to adults that start in childhood. And then there are adverse community experiences such as poverty and violence. And these happen at the built community level. And then there are atrocious cultural experiences that have to do with historical trauma from slavery, colonization, forced family separation in some situations, and harmful social norms. So ACEs actually go beyond adverse childhood experiences. And when you put all of these together, and we look at substance use, and we look at family cohesion, we can see how things can be dislocated, dislodged, and actually appear as confetti that's coming down and you don't know what to grab on first. But I think that this discussion will give us a foundation where we can begin to understand the several different layers that exist in our population, where if we take the proper steps, that we can actually prevent some of the harms that we've talked about today. So in terms of adverse childhood experiences, we're talking about 10 things that could happen during someone's childhood before the age of 18 is what the research shows. So if there is abuse, physical, emotional, sexual, neglect, physical, emotional, household dysfunction, mental illness in the home, mother treated violently or father treated violently, divorce, incarcerated relative, or substance use, if 4 or more are experienced before someone's 18th birthday, then there's some severe outcomes that could occur during adulthood. If there are 6 or more, then that could suggest that there are 20 years less life expectancy. So it's a very serious matter. The brain forgets, but the body remembers. So stored in our DNA, we have the trauma that can occur from these experiences. And it doesn't matter which 4, and it doesn't matter which 6. The study was done with about 15,000, I believe, primarily white, middle-class individuals. And so this is not a matter of ethnicity. It can happen to any of us. And it's interesting that the study was done with white individuals. And now added to the ACEs is a little icon that's racism. So the impact of racism on Black and Brown people is also an added ACE, where there's another level of a challenge that can exist. And these are the possible outcomes. And as you look at this, I want you to think about the kinds of things that we want to prevent. So I've coined a phrase in Together for Resilient Youth that's called transformative prevention. In transformative prevention, we don't just look at programs and initiatives in a silo. We want to make sure that we're using the public health model, which means, of course, that we're looking at root causes. And so when we talk about prevention globally, we can actually look at adverse experiences and link them to the kinds of outcomes that we want to prevent, such as lack of physical activity, smoking, alcoholism, drug use, missed work, severe obesity, diabetes, depression, suicide attempts, STDs, heart disease, cancer, stroke, COPD, and broken bones, which stands for violence. So if we really want to get at the issue and get at the challenge and get at the root cause, then we ask the question, not what did you do? we ask the question or seek to find out what happened to you. Over 6,000 individuals were part of focus groups and discussions about adverse experiences, including all ethnicities, either separately or in a group together, young people 18 years of age and older, a Latinx group, only Spanish-speaking individuals, and the result was the same. Not only did a community evolve in terms of respect and understanding and thoughts about how we communicate and how we see each other. So out of that has sprung what I call the resilient community. And then we, of course, look at equality and equity. So it's not good enough to provide everyone with a bicycle that doesn't fit their particular need. Although they all have wheels, and that's equality, then we kind of think that we've done the best that we can do. But when we look at equity, we seek to make sure that everyone has the same wheels in terms of wheels, but there's an adjustment in terms of what fits for that particular person. And as we're talking about it, as that particular group of individuals. ACEs can predict earlier age of onset, drinking onset, prevalence ratios for current and ever smoking have been shown to increase as ACE scores increase. In prescription drug use, for every additional ACE score, the rate of number of prescription drugs used increased by 62%. Each ACE increased the likelihood of early initiation into illicit drug use by 2 to 4-fold. So as we're thinking about what to do about marijuana, what to do about prescription drug use, again, foundationally, we need to ask, what happened to you, not what did you do? These are some of the outcomes of trauma that I won't go into. This will be available to you. Thinking, forgetfulness, emotional, depression, physical, rapid heart rate, sleep problems, spiritual hopelessness, in behavior, isolation, violent behaviors, in relationships, angry at others, isolation. Now, some of the ACEs, as you saw in COVID-19, in terms of isolation, how many folks have experienced being angry with family members or a spouse? And trauma doesn't need to be 4 or more of those things. There are other traumas and challenges that happen in our lives that can stop us from moving forward to be the best that we can be. And resilience. Resilience, I feel positive about my future and what things impact that view. Why am I not positive about my future? What can I do in order to be more positive about my future? What is my resilience, in my resilience toolkit that will allow me to continue on successfully in life? And so, speaking of toolkit, that's one of the things that Together for Resilient Youth worked on with the Duke School of Nursing and CTSI to create a toolkit from those 6,000 individuals, but more so on the focus groups that were held, so that they would communicate to us what resilience looked like for them. And many times, treatment is not necessary through counseling or psychotherapy. However, if one needs and wants that, It's an absolutely wonderful way to go. But there are interventions that can occur without that if someone wants to look at the toolkit and determine those outcomes. And the other important things, and the reason that I'm mentioning these is because we can use these in order to respond to what ACEs presents us with. One is shredding negative thoughts, not paying attention to everything that goes through our mind. And we use that very often with young people and with young adults and with adults. But specifically in terms of some of the decisions that young people make, who are you listening to in your head? Or who are you listening to in your public life among your peers? So the questions are, what or who uplifts you? What or who gives you strength? What or who helps you get through hard times? You may have a what, you should have a who, because every child, and I believe every adult, needs at least one adult who is irrationally crazy about him or her. Of course, this is an entire training and session that we don't have time for today. So in terms of prevention, the conventional thought in terms of how to respond to people that are exercising some of those challenges that we saw after ACEs, people are bad, people need to be punished, we need to stop making excuses for people. What is wrong with you? And we saw that during the discussion with the young people. Is this the kind of conventional thinking that's happening? And then there's trauma-informed. We have evolved to trauma-informed, which is a good thing. People are suffering. People need an effective intervention, singular. We need to focus trauma impacts on development, and that's what happened to you. So that's important. But then in transformative prevention, it's resiliency informed, which means that people are resilient. People need our compassion as they learn new skills. We need to learn how skills of well-being can reduce suffering. And then what is right with you? What are your strengths? This is about the social determinants of health, and it speaks to communities that work together and are resilient together can assist each other to wellness. So the social determinants of health, very quickly, there are 5 buckets in the social determinants of health: economic, stability, education, health, and healthcare, neighborhood and built environment, social and community context. And as you can see the picture evolving from the tree now here, there are communities in Durham and in every county, state across the United States that have those pockets of low wealth, low resources, where we tend to think it's the people and we don't look at the built community. And so now we'll go into talking about coping together and how this wonderful partnership has developed in order to help families in their search for resilience.
Thank you, Dr. Boone, and thank you for inviting me to co-present with you. I am Eve Puffer. I'm an assistant professor at Duke in the Department of Psychology and then in the Duke Global Health Institute. And Dr. Boone and her community health workers that she has trained have given us the opportunity to partner with them to co-develop and co-deliver this program called Coping Together. And you'll see here that they're the community health workers. Thank you. These are the community health workers who delivered this program. And you could see on the slide before, it does say from Kenya to North Carolina. And the reason for that is that before COVID-19, all of my work on family interventions was in Kenya. And so we were able during the pandemic to find opportunities to actually adapt some of those approaches to North Carolina. So it's a direction you don't see often from lower middle income countries to high income settings, but we were given the opportunity to do that here. You can go to the next slide. So the COVID-19 pandemic raised awareness of the mental health disparities that we have here in the United States, and also, of course, disrupted our social and mental health services. But it's important to know that, this is overlaid on an already staggering mental health treatment gap in the United States. So the COVID-19 pandemic raised awareness around this, a problem that was already there. Next slide. So we show here a guiding theory of change that if we are to improve family functioning through family-based interventions, we hypothesize that we can improve child and caregiver mental health, preventing disorders or treating them. And we define mental health very broadly. So I am excited to talk with you all so that you can help us think about how that also relates to substance use. Next slide. So DGHI provided funding for our group at Duke to partner with community partner organizations to do this adaptation process from the Kenyan intervention. So we quickly identified TRI as one of the most active and well-fitting community partners for our for this program. You can go to the next slide. So what is Coping Together? Coping Together is an 8-session intervention. It is virtual. So we delivered it via Zoom with a facilitator who was trained in the program, and that was a community health worker. But the community health workers themselves were also part of developing these 8 sessions. So you can see that the program starts with discussing family values, communication skills, creating family vision and a family plan. And the second half gets more into details of applying those skills to family-specific challenges, problem-solving skills, coping skills, conflict resolution, and then solution-focused action planning, and then their graduation for the program. And you can show the next slide, Dr. Boone. This is one of our central metaphors, which is depicting the family as a tree. The trunk is the caregiver or caregivers, leaves are children, and then the branches connecting those, symbolizing the relationships. But importantly, we emphasize the roots, which are sources of support for that family, which differs by family, but can be values, culture, faith, beliefs, and members of their extended family and community. So on this next slide, you see one of the central tenets of the program, which is that all families have to endure storms, many of which we cannot control, and that the skills that we learn and work on together and coping together can help us to become resilient even in the face of those storms. You can go to the next one. So this shows that we had 5 community health workers. We did a small pilot study with 6 Coping Together groups who met weekly with these CHWs. So we want to briefly walk you through some of the preliminary family outcomes that we had from our pilot, and we'll focus on the qualitative results. So first, if we can go to the first result, we looked at overall family closeness. And this is what Dr. Boone has been talking about family cohesion as a protective factor. And we did qualitative interviews. One caregiver said we talked about being kinder to each other, and the boys, this caregiver had 2 foster boys, have really been working on it. I think the whole house is a little quieter, gentler or something. And then in the next slide, we see caregiver youth communication, which was one of the main target outcomes. And youth described more positive communication. One youth said that after learning communication skills, they were starting to tell their caregiver more of what was going on in their head. And then the youth was saying that it used to be conversations were just about school or mundane things, but now it's more positive, talking about accomplishments or just more positive conversation. And then here you can see it depicted in a different way. We asked families to draw their before coping together self and their after coping together families, and then to explain that in the youth focus group and interviews. And this youth showed that they started off on not good communication terms, and then that they grew closer together. We also asked youth to do an activity where they wrote a letter to their before coping together self to tell themselves what they would be learning. And this sixth grader said, you'll learn communication skills, how to express yourself. You'll also learn how to listen to others and grow closer to your family. So in that response, showed that some of our theory of change of what we were trying to change did lead to a perception of that change from the youth's perspective as well. And a ninth grader wrote, "Dear old me, you are about to learn how not to ignore your problems. High school is tough, but you don't need to care, worry about others' opinions. You become more confident and face your problems head on. This helps with growing to do things you thought you wouldn't be able to do, gives you confidence to do those things." And this, I think, resonates with TRI's overall mission as we were looking at the results and how coping together can hopefully fit into the overall idea of transformative prevention in TRI's suite of programs. I think we'll just end with this. This is a quote from a caregiver in which they connected our metaphor of the tree to their perceived changes, saying some families aren't close, but now our tree is full of leaves. It's beautiful. It has green leaves and it has a strong trunk, a strong base, and it's rooted into the ground. So we are excited. Dr. Boone has led efforts to start more rounds of coping together. So we'll continue to do research on how this could be rolled out into the community, and then how we can also conduct larger research evaluations to really develop a stronger evidence base so that we can scale up. So here's our contact information. I know you all have Dr. Boone's, mine as well, and happy to connect with anyone.
Thank you so much. I am really excited about what we presented. And as we head into the lunchtime, I think we have 15 minutes to grab something, but at least don't forget to come back because there's more in store for you. Really excited for what we've learned this morning and what will come this afternoon. So thank you so much. So I have the pleasure of introducing Dr. Seronda Robinson, who is the chair of the Department of Public Health Education at North Carolina Central University. That department offers a professional preparation program. The mission of the Department of Public Education is to prepare students academically and professionally to assume leadership positions in public health. The department provides students with opportunities to develop proficiency in the application of theory, content, and skills to promote, support, and enable healthy communities. The public health education program is designed to prepare students as entry-level health educators for practice in a variety of settings, such as public health departments, voluntary health agencies, healthcare organizations, corporate work sites, and community-based organizations like TRI. Thank you so much and welcome, Dr. Robinson.
Thank you, Dr. Boone. I'm only going to be before you for a short period of time to present the campus perspective on marijuana because the students have already done such an excellent job of presenting that perspective today. So a little bit about marijuana and college students. So marijuana is the most frequently used illicit drug in the United States and on college campuses, with many students initiating it their freshman year. Initially, there was a rise in the use of marijuana in college students between the years 2015 to 2020, but then we saw a decrease in 2021 as the pandemic continued. So we need to conduct further studies to determine what led to a decrease in use. Was it related to the availability of the drug? Was it related to family involvement, differences in peer pressure? So we need to try to find out what could have led to that decrease so we can harness it for our future preventions. Some problems associated with marijuana use are social and behavioral problems such as isolation, poor academic performance, violence, and crime. Of course, we know marijuana is a gateway drug, and nearly all adolescents and teens who use other illicit drugs also use marijuana. Now, for those who use marijuana, they're also engaged in other high-risk behaviors. They tend to be more likely to use alcohol or tobacco, to engage in drinking or use drugs during intercourse. They spend more time at parties and socializing and less time studying. And they have a lower perceived importance for religion and community service. Now, what are some of the reasons that college students use marijuana? And the students already told you it was to socialize, or a way to have something in common with others, or a way to get prepared for the game or to celebrate. But they also do it because of curiosity. Like I say, when they get away from home and they're unsupervised, they become curious about trying new things. Some do it due to peer pressure. They see other students around them engaging in it, and they often are curious or influenced by their peers to try it as well. Some students engage to deal with boredom. We found that a lot of our students don't know how to entertain themselves, so they don't know what to do between classes or on the weekends. And so some studies have shown that they find the best way to pass time is by using marijuana. There is a sense of perceived safety. The students don't seem to realize the dangers involved in using marijuana. Some use it as a way to escape life challenges. They don't know how to deal with the pressures that they face or to deal with the problems encountered. We know now a lot of students are coping with these changes in not socializing with peers and being more virtual. And so they use this numbness to kind of escape challenges. Some also use it to manage their mental illnesses. So it kind of helps them deal with anxiety or depression, or even with post-traumatic stress syndrome. Some do it just for that feeling of getting high. You know, they want to feel that inhibition so that they are more comfortable socializing with people and they laugh more easily and engage more easily. And some do it just because it's easily accessible. But what are the reasons that other students choose to refrain? Some students just have no desire for the effects that it has on them. And some are concerned about their health because, as we know, it has long-term health effects. And then there are students who feel that it's against their religious or moral beliefs. But then when we think about the effects of marijuana, it has significant implications on academic success. Because when students have altered motivation and cognition, it makes it difficult for them to acquire new information. So they have a hard time learning in their classrooms. They have an altered sense of time and decreased reaction time. So if they're called on, they may have a decrease in the time that they actually use to respond. They have difficulty listening and speaking, or impaired or reduced comprehension. So the teachers are talking and they're not necessarily comprehending what's being said. And it impairs their short-term memory. So they may not be able to respond or to even do the homework after they just learned the topic in class. They have impaired learning and memory perception. They have impaired judgment and problem solving. And of course, those are all the things that are critical to performing academically. While some are using it to deal with anxiety, it can lead to intense anxiety or panic attacks. It can even lead to paranoia. Some develop a psychological dependence on it, or they may experience withdrawal symptoms if they try to pull away from it. There's a reduced ability to perform tasks that require concentration and coordination, and a decreased ability to deal with the stress that often accompanies personal growth and achievement. And that's the very reasons why students come to school, to grow and to achieve greater things. Some of the first signs that we should be aware of to recognize substance use among students is procrastination and lack of follow through. So are they waiting to the last minute to begin their assignments? Are they not completing them? Are they not turning assignments in on time? And there may be a decrease in grades and an increase in missed classes. Some recommendations for dealing with this, if we notice these signs and symptoms, we need to help the students to cope with stress and time and work management. We also need to target prevention messages to groups who are at higher risk, such as freshmen, athletes, or Greek sororities and fraternities. We should also involve our students in prevention and education efforts, because we do want to make sure that our messages and our tactics are culturally appropriate. And also when the population, your target population, is involved, they may be more likely to engage in the success of these efforts. We should train our faculty, staff, and students to recognize the signs and symptoms. And we need to make our policies clear to parents and incoming students. And of course, we should engage with the efforts of College Try. Now, there are limited studies on the particular impact of marijuana use on HBCU campuses, but those that have been conducted suggest that some of the things that may decrease the use of marijuana use on HBCU campuses as opposed to other campuses are the strong focus on character development and religious expression that is instilled in students on HBCU campuses. Our campuses also present a nurturing, supportive family environment, as our last study just showed the importance of family. And as our students reported, we have strict enforcement of our policies. And so those are just some of the things that kind of decrease the use on campuses. So of course, we see some marijuana use, and as the students indicated, some students try to mass use through e-cigs, but then we do take these steps to reduce or limit the use on our campus. There are my references. And I think we're going to take questions after the next presenter, but if you have some, and it's okay to ask at this time, I'm available, and there's my contact information. So thank you very much for your time.
Thank you, Seronda. That was a marvelous presentation. And so now we're going to hear from another voice of administrator who is involved in dealing with marijuana in a campus setting, and that's Tom Szigethy, who is Associate Dean of Students and Director of the DUWell Center here at Duke University. And Tom's going to talk to us about the efforts at Duke in terms of marijuana and other drugs of abuse. Tom?
Thank you, Ed. And I'm just going to talk a little bit about the culture at Duke. I appreciate what the students had to say, and I agree with them in a lot of aspects and a lot of ways they were presenting. What we have found, if we look at our data over the years, about a decade ago, we had around 14% of students that were smoking marijuana. As of 2020, we're up to 23%. So it's definitely increasing, similar to what Seronda had just presented. And what we're finding is across the nation that it has increased. And a lot of that really comes down to how many states are legalizing it. The issue that we find amongst students as they come through to our office and discussions if they've gotten sanctioned or in trouble for smoking marijuana is that the legalization gives them the impression that it's not as serious of a drug, which is a very difficult thing to combat at times. Although if we look at nicotine and alcohol, kill the most people every year, more than all other drugs combined. And those are the two legal ones. So the biggest risk with the legality of it that I find is the prevalence and how easy it is for students to get their hands on the drug. As our own students talked about, that they haven't heard a lot around pot. We've done obviously a lot around alcohol. Our students, as they walk in the door, 70% of them are reporting that they don't use substances at all. And then by October, 70% have experimented, with around 20% being in that high risk category. So we've done an enormous amount of work around the alcohol messaging through orientation in order to get people to reduce that risk of obvious accident and death, which is very highly likely with the alcohol if they really are not doing so in moderation. The overall message on campus tends to be one of harm reduction. So if students are sanctioned or cited as violating policy, then they're brought through an educational harm reduction process in order to learn what went wrong and how to manage this, even though the recommendation obviously is to follow the law and not to use. But if they are going to drink, if they're going to smoke pot, how do they do so in a way that's going to put them at the least amount of risk? And with the pot, it brings on other implications. It's difficult for staff in the res halls to always to locate who it is that's potentially smoking. If a floor smells like pot, to try to narrow it down to the actual specific room is one of the challenges. There is some workshops that we've put together. We have a brand new one last semester, had 40 freshmen come through that on the different illicit drugs that are out there and what the risk factors are. As many of you have probably read in the paper, around Greek life has shifted dramatically in the past year at Duke. And that used to be the source where each chapter would come through different education over the years. And it was a very easy way to get a large percentage of students trained. And that's not the case now. So what we're really looking at is new and inventive ways to try to get the information out to the students, since we don't necessarily have student organizations coming in voluntarily saying that they want to know about this information. And since we're not at a high school or a middle school, we can't necessarily mandate it. So the reasons that we find that students are using a huge piece, which has been mentioned throughout the day, is social engagement. Dr. Parnell had mentioned that stamina or the social abilities tend to be minimized after using, and yet our students seem to be using it in order to collect friends or to be around friends because of the simple fact that they'll smoke together. We have found a slight increase since the pandemic has hit in our survey data, from about 21% to 23% who are using pot. So that's different than Central and what Dr. Robinson had just presented. And the assumption there really is that with the alcohol, if people were going out to parties in the neighborhoods, it was very obvious that they were breaking COVID rules and they were getting reported pretty easily. But if you have a small group of people that collect in a room, it's a lot more difficult to identify them and to have them get in trouble for anything besides that in regards to COVID and violations and mask use and those kind of components. We also have about 51% of our students are coming from areas that have legalized pot, and that through either medical or medicinal use, as well as just use in general. So they're able, when it comes to sourcing and acquiring the pot, often they'll get it from home in regards to their hometown or the contacts they might have, and bring it with them when they come to campus. So overall, it's an ongoing battle in general, as we all know, in regards to prevention and getting information out there and getting the word out. Some fantastic information today that we will be utilizing as well in order to beef up our prevention efforts on campus. Thanks.
Okay, thank you. Thank you, Tom. Thank you, Seronda. So we do have some time for a bit of discussion if anyone has any questions or topics they'd like to bring up. I just wanted to kick it off a bit. So like with alcohol, I don't know if we're going to be able to, completely suppress it or combat it. Is there a strategy of kind of managing it and maybe identifying individuals who may be getting into more habitual, troublesome use? What do you think?
I think yes. I think a lot of this becomes shaping the community, as we've been doing for numerous substances in general, but trying to get the students to recognize that the goal is not to get high. The goal is to respect everyone else in the community. To bring one of the efforts really is to bring a whole dorm floor together when they're smoking in a dorm and it's getting in the ventilation system and everybody's smelling. It has an impact in the entire community. So the community itself can begin to put pressure on the students who are actually smoking.
Huh. Yeah, I think so.
I agree. We have to educate the students, as I mentioned earlier, from before they come in and continuously deliver that message, that is hazardous not only to their health, but to everyone's health around them. And then just the impact that it has on the physical health, the mental health, and that it can lead to, lack of success. And that's counter to their reasons for even being on campus.
Mm-hmm. And in terms of the association of use of marijuana with other illicit drugs, I mean, is that something that you've seen occur? And that's a big problem on the campus?
I think with us, marijuana and alcohol are the two most used drugs. We don't see many other drugs used there. Of course, they have the e-cigs and they have various things in them, but we haven't necessarily identified what is contained in them. So marijuana and alcohol are the two that we know about.
Okay.
And I would agree at Duke as well. It's marijuana and pot or marijuana and alcohol are definitely the top two drugs that are being used. And I think the marijuana oftentimes is being used, at least in early stages, to try to mask anxiety and social discomfort until it grows to a point where it might be exacerbating some of that behavior. And then the students are interested in trying to address it.
I see Diana has her virtual hand raised. Diana, you want to unmute?
Didn't make it. Okay, there. Yes, I just wanted to bring up, there was a really nice article that was recently published about lower risk cannabis use guidelines for reducing health harms from non-medical cannabis use, published in the International Journal of Drug Policy earlier this year, the lead author being Fisher, Fischer, initial B, a whole series of authors, but the next one is Robinson, Robinson, the initial T. So if you put that in PubMed, this should pop up. It has some really great guidelines for how to talk about reducing harms associated with marijuana use.
Great. Well, thank you, Diana. That sounds valuable. Now, I have a couple of questions in the Q&A. Connor O'Neill says it'd be curious to learn about the potential trends in legal cannabis products such as Delta H, THC, in college populations. Do you have any information about that? And people vaping these products?
I don't have direct numbers. People are definitely vaping on campus and around town, but we don't have the actual survey numbers on that.
Okay. Is there, in terms of vaping at NCCU, is that a more predominant way recently rather than traditional smoking? Yeah.
Yes, we have seen that, an increase in vaping.
Okay. And then Robin Huffman says North Carolina legislatures are not hearing this story about the research and science and impact on health and on youth, especially research that shows that cannabis worsens mental health conditions. And it's assumed that legalization will pass. And I think that's probably the case. But I mean, like alcohol, alcohol is legal within constraints, but it still is a problem. And in terms of the kind of psychology clinic that deals with mental health issues among the students at Duke and at NCCU, is sometimes drug use an aggravating factor. I would think that might be something that would be attended to in terms of kids who are starting to get into trouble with their drug use.
Yeah, and Jason Kilmer had done research on this as well and reported that what they're finding in adolescents who are using pot regularly is that they can present with a lot of mental health issues. To get them off of the pot, you cannot necessarily accurately diagnose them.
Okay, great. And so, another thing from Jeff Georgie, some students use THC as medication for ADHD. And there is a lot of information and misinformation out there and things that are not well grounded in research that, because marijuana is the color green, then it must be good, right? So that they're, I would think that, having counter information that maybe it's not always medicinal might be a good thing to convey. So, okay, so we do have some time now, unless there are any other questions. Well, Wanda, you have your virtual hand up.
Yes, there are just two things that I wanted to mention, and one is the danger of marijuana being laced with fentanyl.
Oh.
It is extremely dangerous and very pervasive. And so that is of concern. And we see that information in the opioid overdoses. That is another area of study that we do, and lots of people are studying that. And that is a real concern. And so we need to look out for that on our campuses. Also, Attorney General Josh Stein will be talking about marijuana and vaping when he does his presentation. So I just wanted to mention that. Oh, the other thing is the misdemeanor diversion program with the Durham County Juvenile Justice Center. The misdemeanor diversion program covers young people ages 16 to 21, and they're trying to expand it otherwise upward. And so if one is cited for alcohol or marijuana or whatever the case may be, as long as it's not a gun crime or something that's not a misdemeanor, then they can go through this short program. And by short, I mean 30 to 60 days of their choosing, and then nothing will hit their record. So, I would like to propose that in any situation. So, and, I can talk about that offline later. Thank you.
Yeah, thanks. And then you bring up a good point in terms of the adulterated cannabis and the real dangers of that. And maybe that's one positive feature of legalization. And if there are some controlled dispensaries like there are in some states, then you could be more confident of the purity of the product. Nicole, you have your hand up?
Yeah, I'm still going back to what the students were struck by about the difference in enforcement between our two campuses, between Duke and NCCU, and you even emphasized that, Dr. Robinson. I'm wondering if there is tracking of outcomes. So both on the Duke side and on the NCCU side, like the students who are getting caught and going through intensive education at Duke versus intensive enforcement at NCCU, what are the outcomes, and is behavioral change occurring, and are those students going on to thrive and graduate? How do the choices impact the students?
I'm not aware of the particular tracking. I'm sure they have that data in our Office of Student Conduct, but it would be interesting to compare the two, like you said, with the strictness that we have versus what they go through at Duke. Of course, there were times like a first offense, they may receive those workshops that you mentioned. So we do have those type of, harm reduction and prevention workshops. But as the students indicated, if they are caught with it in the residence halls, then they can lose their housing. So it's definitely not allowed in the residence halls.
Interesting.
And I would agree as well. We don't necessarily have numbers to track that, although it would be a very interesting piece to track. I also don't want to give the impression that Duke does not do anything, because the police do get called in, and the police will give citations to students if that is warranted. They're not incredible. It's not to the degree where I would say they're as strict as the Central students are saying. It's very clear, it's very pervasive, everybody's very aware of it, and that's clearly not the case at Duke. But we know on the risk mitigation, at least around alcohol, that we have 3% recidivism through the process of education. So I'm assuming that we're going to have somewhat of that with the pot, although I will say that the students coming through who smoke pot are much more loyal to the cause. The students coming through with alcohol are very interested to figure out, what did I do wrong and how do I do it safely? Pot students tend to be, and I know I'm generalizing here, but they tend to be very well researched on their craft, and they have every single argument and defense to defend the behavior. Yeah. And they're not as interested in changing behavior as the students who might be on the high-risk side of alcohol use.
I see. Okay, well, thank you. We have a couple more comments in the Q&A. Grace Marsh mentions that the misdemeanor diversion program has already been updated to 18 to 26. So those are the age range for that. And then Robin Huffman again says, I hope we can make a distinction between decriminalization of cannabis and legalization. So that certainly is important. One is, that there's no stiff penalty, maybe just a traffic ticket sort of citation. And the other is that there are actually dispensaries. And and then here's one from Diana Lazowski. To the contrary, studies in California have shown that THC products obtained through dispensaries are also adulterated. With molds and fungi and much as 50% of the product. So there's no significant, this is so significant that oncologists are warning that immunocompromised patients refrain, and some have died from microbial infections. Dispensaries do not equal quality, and this is not a pharmaceutical product. Those are excellent points. And so we need to be aware of that. And other things in terms of pesticides in the product and so forth. And the other that's been brought up earlier is in terms of just the percent THC in the product. And, back 50 years ago, it may be, or was quite a bit less potent than it is now. And the idea that, people don't die of cannabis overdoses, well, maybe until they get behind the wheel of a car, maybe. But it can lead to other things that are self-destructive. So, okay, well, any other comments or questions? If not,
Yeah, I just wanted to say
Yeah.
that the balance of CBD to THC is really what has changed. It's almost like the CBD was a protective factor in marijuana, but the tetrahydroxycannabinoid is what has shifted. So the balance has shifted, and therefore the dangers that are associated with marijuana in the modern form.
Yeah, great point. And in some senses, it does CBD does counter some of the THC effects, and CBD is sold widely. I mean, I just a half a block down from my lab, you can buy CBD in a lot of different forms. So now we do have a message from our Attorney General, and I think we have a time here to go ahead and have that. It's a pre-recorded message, but he's been very on top of the issue. And Wanda, do you want to
introduce him? Yeah, I'll introduce him, even though he's not here. So Josh Stein was sworn in as North Carolina's 50th Attorney General in 2017. The Attorney General is elected by the people of North Carolina every 4 years as the state's top law enforcement officer and top lawyer. Attorney General Stein is focused on protecting North Carolina families from crime and consumer fraud. Attorney General Stein has also made combating the opioid crisis as a top priority, and also vaping. As the head of the Department of Justice, the Attorney General oversees criminal appeals from state courts to ensure that criminals are kept behind bars and innocent people are not. Stein also ensures that consumers are protected by going after scam artists and corporate bad actors. The Attorney General protects the taxpayers as well by providing legal representation to state government agencies, departments, and commissions, and by investigating and cracking down on Medicaid fraud. I just want to also say that there is a lawsuit that the Attorney General will probably speak about here in terms of opioids and a bellwether case in Durham. So thank you so much, Attorney Josh Stein. We're used to you being here in person, but we look forward to hearing what you say in your video. Thank you.
Hi, I'm Attorney General Josh Stein. I'm pleased to be able to join you all today, even if it's just by video. As your Attorney General, it's my responsibility to protect the people of North Carolina. And job one for me and for us all is protecting our kids. That includes protecting our kids from addiction and the dangers of substance abuse. I'm grateful to the Duke Center on Addiction and Behavioral Change and Together for Resilient Youth for their leadership on this issue. And I'm glad to be able to join you all today to share a little bit about my work to keep our kids safe from addiction, from violence, and from unfair treatment, and give them every opportunity to build stable, successful futures. Now, I know that the focus of this conference is marijuana, but before I get there, I want to talk to you a little bit about the breadth of the issue of addiction and how it impacts our kids, starting with e-cigarettes. A few years ago, I started noticing that JUWell pods were being left in my TV room at home. I didn't even know what they were. So I asked my boys, and they told me, they were teenagers at the time, that what e-cigs were, what JUWell pods were, and how so many of their friends were using them and simply just discarding the pods all over the place. Then a couple weeks later, within a week of each other, I had a couple of my buddies tell me about their kids, 9th graders at the time, dependence on nicotine through e-cigarettes. The kids had become addicted, their grades plummeted, they had behavioral issues, they had to change schools, and they needed medical intervention for their addiction. The root of this problem? Slick advertising campaigns on social media that highlighted kid-friendly dessert and fruit flavors delivered in a sleek, concealable USB device. The toxic marketing claims used to peddle e-cigarettes to America's young people had a devastating effect. They had undone years of hard public health work because in the late 90s, nearly 30% of teens used some form of tobacco products. By 2017, we had driven that number down to lower than 5% of high school students. But thanks to JUWell, those hard-won gains had evaporated like a puff of vapor. Nearly a quarter of our young people now use some form of nicotine every day. In 2019, I filed the first state lawsuit against JUWell, the 800-pound gorilla of the e-cigarette market. I alleged that JUWell designed, marketed, and sold its e-cigs in a manner that targeted young people. Last June, North Carolina became the first state in the nation to hold JUWell accountable. My office entered into a consent judgment that forces JUWell to fundamentally change its marketing and sales practices to protect kids and to pay North Carolina $40 million to help young people conquer their addiction to nicotine. My hope is this agreement keeps JUWell's devices out of our kids' hands, its chemical aerosol out of their lungs, and its nicotine from poisoning and addicting their brains. But the huge market of young people addicted to e-cigarettes that JUWell helped to create didn't just disappear when JUWell started playing by the rules. Other companies are filling that vacuum. That's why I'm investigating companies in the e-cigarette supply chain, from manufacturers to distributors to retailers. But ultimately, this game of whack-a-mole is untenable. We need the Federal Drug Administration to step up to prohibit nicotine manufacturers and retailers from marketing and selling to young people, using youth-friendly flavors, and developing high concentrations of nicotine that accelerate a person's dependence to that product. I've led a national coalition of attorneys general to write the FDA, urging it to protect kids by imposing these types of restrictions on the entire industry, and I will continue to lead on this issue. But just as we have to prevent our next generation from getting hooked on e-cigarettes, we also have to protect our kids from marijuana. With so many states legalizing marijuana, North Carolina is looking at medical marijuana legislation. So we have to have a broad conversation about marijuana that considers its public health, racial justice, and economic implications, among others. Let's start first with the issue of racial equity, because the facts are beyond dispute. White and Black North Carolinians use marijuana at very similar rates, yet Black people are disproportionately arrested and sentenced. In 2019, there were approximately 30,000 charges and 8,500 convictions for possession of up to a half an ounce of marijuana, which is a Class 3 misdemeanor. 61% of those arrested were non-white. The disparities are very similar among those who were convicted of the charges as well. This inequity led the Task Force for Racial Equity and Criminal Justice, which I'm honored to co-chair, to study marijuana policy and make a number of recommendations. The task force, as you may know, was formed in June of 2020 to take a hard look at the ways in which our justice system fails Black people and other people of color, including young people. We came up with 125 separate recommendations to help create fair law enforcement and court systems and to promote public safety. One recommendation is to deprioritize marijuana-related arrests and prosecutions. A second recommendation is for the General Assembly to decriminalize possession of up to 1.5 ounces of marijuana. And we also need to expunge the criminal records of folks who were convicted of this offense in the past so that they can get on with their lives without a criminal conviction getting in their way. I believe the time for decriminalization of marijuana possession is here. The racial disparities are unacceptable, and we must act. A third marijuana-related recommendation is to convene a task force of stakeholders free from conflict of interest to study the pros and cons and options for legalization of possession and cultivation and or sale of marijuana, including by government or not-for-profit monopoly options. The study should be guided by a public health, public safety, and racial equity lens. While I continue to believe a broad-based task force is the best way to go, another conversation has been jump-started by the medical marijuana legislation that's currently pending in the North Carolina Senate, sponsored by Senator Rabin. I support medical marijuana, yet I do have some concerns about the legislation. To his credit, Senator Rabin has expressed a willingness to work with me and other stakeholders to improve his bill. Here's what I have told him. In other states, we have seen that the legalization of medical cannabis leads inevitably to adult cannabis use. If we're going to legalize medical marijuana in North Carolina, we need to build a regulatory framework that can eventually handle adult use marijuana as well. There needs to be a single independent authority to regulate all cannabinoids, including those currently being sold in convenience stores right outside our public high schools today, Delta 8 and Delta 9 included. If the state legalizes marijuana sales, it is important that retailers not be incented to maximize sales. For this reason, a state-directed monopoly to control all retail sales of marijuana, much like our current ABC system for alcohol, is a good idea. With alcohol sales, North Carolina has among the lowest liquor per capita sales in the Southeast and among the highest per capita tax revenues. To me, that is the ideal outcome. We also need strict regulation of marijuana products, marketing, advertising, and the density of marijuana retail outlets. We need to not only capture substantial tax revenue, but dedicate a defined portion of it for prevention, treatment, enforcement, research, and other strategies that pursue public health, public safety, and equity goals. These are among the top line recommendations I have made to Senator Rabin. Whatever path our state follows, I hope we can all work together to ensure that any marijuana policy furthers important public health, public safety, and racial equity goals. Because we are talking about racial equity and the well-being of young people in North Carolina, I'd like to touch on some of the other relevant topics addressed by the Task Force for Racial Equity in Criminal Justice. Each interaction with the criminal justice system can have a negative long-term impact on a child's development, even affecting them into adulthood. We have to be honest about the racial disparities that exist in school discipline and juvenile justice. Young people of color are overrepresented in the juvenile justice system. Children of color make up 46% of the population aged 6 to 17 in North Carolina, but made up 71% of complaints received and 80% of commitments to youth development centers. We absolutely must keep our schools safe and respond to student behavioral issues. But we can do that in many instances without taking kids out of school and putting them into the criminal justice system. It's not good for them, and it's not good for our communities. An entire section of the task force's recommendations address the topic of school resource officers, or SROs, the law enforcement officers who are detailed to our public schools. SROs exist to keep kids and teachers safe and to build relationships with students and school personnel. Sometimes SROs should refer students to the juvenile criminal system, but in most instances, such referrals can be counterproductive. School personnel should also, whenever possible, be focused on keeping kids in school and addressing their social and emotional needs, as well as their academic ones. That's why we recommend that SROs and all school personnel receive training on the proper use of SROs so that no adult is unnecessarily referring kids to the juvenile justice system when school-based discipline and restorative practices are sometimes more appropriate. The task force also recommended that communities create school justice partnerships. In these partnerships, a group of community stakeholders develop and implement effective strategies to address student misconduct to reduce the number of suspensions, expulsions, and referral to the justice system. These partnerships have proven to reduce students' juvenile court involvement. We also recommended that schools hire a sufficient number of nurses, counselors, psychologists, and social workers. And then all school personnel complete medical and mental health first aid, cultural competency, and developmental disability training. These professionals are often best equipped to help our young people when they're facing challenges, and they're also the most likely to recognize the signs of issues like substance misuse and addiction. Finally, we also recommended that data be collected on discipline in schools. In fact, the School Safety Action Plan and the Task Force for Racial Equity and Criminal Justice have several recommendations that focus on collecting and sharing more data. Because by collecting more data and making it public, it will better inform our work so that communities have more confidence in the policies that we pursue. The issues that you all are discussing today at today's conference are urgent, and I want to thank you all for your commitment to North Carolina's children. It's on all of us to keep our kids safe and to put in place the protections that they need to ensure they become successful adults. And when they're struggling, we must intervene quickly and give them the resources they need. The conversations around marijuana decriminalization and legalization are getting louder. And as they happen, we must focus on any unintended consequences to our children. I am committed to doing everything in my power to do right by our kids. And the Department of Justice and I look forward to working arm in arm with all of you as we continue this work and build a North Carolina that is safer, stronger, and healthier for all. Thank you.
And that was a very comprehensive talk about not only the hazards of marijuana and how to deal with them, but also nicotine and tobacco, and by extension, alcohol and other drugs as well. How to keep, especially our kids, from harm without doing harm to them. So speaking of potential decriminalization and legalization and our possible future experience with that here in North Carolina, we might take some lessons from the states where that's already occurred. And for one of the first states to decriminalize and legalize marijuana was Colorado. So now we have the privilege of hearing from Elyse Contreras, who's program manager and epidemiologist at the Marijuana Health Monitoring Program and Marijuana Research Grants Program at the Colorado Department of Public Health and Environment. And Elyse has been involved with the Retail Marijuana Public Health Advisory since its inception in 2014 and has worked continuously to distribute the committee's findings and evidence statements and recommendations throughout Colorado and nationally. And now, Elyse.
Thank you, Dr. Levin, and thank you all for having me here today. I am going to attempt to share my screen now. So today I'm going to be talking with you about the Colorado experience with cannabis legalization. This is from the public health point of view in Colorado. My objectives today are to provide just some brief background regarding this legalization in Colorado, and then also to explain our implementation of the public health framework, which is this wheel that you see on the left-hand side. We will go over the assessment strategies, policy development, assurance, and research, and then I will speak to just some of our overall challenges. So to begin with, we will look first at the legalization timeline in Colorado. Medical marijuana was first legalized in the year 2000 through ballot initiative. We then look forward to the 2012 retail marijuana legalization, which in statute, we refer to it as retail marijuana. It is actually adult use or recreational marijuana. This too in itself was a ballot initiative. We then look at a different time scale down here with the medical marijuana commercialization, or the commercialization time scale. Medical marijuana commercialization is when the stores or dispensaries opened in Colorado in 2010 with the intent of serving the medical patient community. That could be arguably the factor that opened the door for retail marijuana commercialization as well, which occurred in 2014. We then look ahead to our timeline in 2018, when, of course, the Farm Bill was passed and hemp, or the cannabidiol or CBD, which can be derived from hemp, began the nationwide legalization. We in Colorado have our marijuana and public health role. This has been defined in statute, but then also different aspects at the public health department, what we do at the public health department in relation to marijuana. First, we house the medical marijuana patient registry. We oversee the scientific advisory panel to review emerging science. We also monitor patterns of use and health effects. We administer prevention campaigns and education. We also have a large laboratory certification and reference library. And we also provide a consultative role to our regulatory agency in the state, which is the Marijuana Enforcement Division. We provide consultation on science-based policies and safety measures. So looking at the public health framework, the first slice of the wheel here in the upper right-hand corner is the assessment, so which we monitor health and diagnose and investigate the issues. So again, as I mentioned, in statute, we are required to monitor changes in drug use pattern and also monitor the emerging science and medical information. And this is exactly how we monitor the health as it relates to marijuana use in the state. So we will first look at that patterns of marijuana use piece and our surveillance of public health data. So we look at both surveys and health incident data sources. So monitoring patterns of use, this is really about who is using, how they are using, how often or how frequent they're using, and are they following safe practices when using. Of note, collecting this type of information and monitoring trends can help focus prevention efforts to target the right population. So these are often the questions that we target in our population-based surveys. Those surveys that we do utilize can be categorized by different populations. So we have an adult survey, the BRFSS or Behavioral Risk Factor Surveillance Survey for adults. We have a Healthy Kids Colorado survey for our youth in grades 6 through 12. We utilize the Pregnancy Risk Factor Surveillance System for new moms and the Child Health Survey, which is a callback survey to the BRFS survey to ask parents who have children between the ages of 1 to 14 living in their household some targeted questions regarding marijuana use. Regarding our health incident data sources, we look to our Colorado Hospital Association data that's looking at discharges with relevant marijuana related billing codes. And then we also look to our Regional Poison Center. So I'm going to just go through really quickly some data from Colorado just to give you an idea of what this looks like. And what I'd like you to keep in mind also are those timelines of commercialization that I went through in our background. So what we're looking at here is the prevalence of past 30-day marijuana use among Colorado adults over time. And what we can look at here, and the red line is from our BRFSS survey. And what we see is that the red line shows an increase of prevalence of use starting in 2017. That's a significant increase, rather, going up to our latest data point there of 18.9% of adults having reported using marijuana in the past 30 days. We look at this in relevant in terms to the US population, which is in purple. As you can see, going back to 2006, marijuana use in Colorado adults has consistently remained higher. And then right around that 2014 time mark is where we start to see an overall increase in use compared to the US average. So consistently, adult use has been higher in Colorado compared to the U.S. For quite a while. Also, demographics in this adult population, 18 to 25-year-olds and 26 to 34-year-olds, the two younger age groups tend to use marijuana the most frequently in the past 30 days. As also males and those who identify as lesbian, gay, or bisexual. And we also see the highest prevalence among white non-Hispanics. Looking now to method of use among adults in Colorado, we see that 3/4 prefer smoking their marijuana, so the 73.7% in the year 2020. Other methods include eating or drinking, vaporizing, or dabbing. I should point out that dabbing is a somewhat newer method of use, which tends to use higher concentrated marijuana products, such as your waxes or highly concentrated oils. That will become interesting when we get to the adolescent section of the data. But what we again see is that the smoking has remained the highest method of use among adults. Here again, we're looking just at use among adults by age. And as I previously mentioned, we see consistently that use among our younger adult age groups remains significantly higher than our older adult age groups. Now looking to adolescent and young adult marijuana use, using our Healthy Kids Colorado data, looking at that compared to the national high school data from the Youth Risk Behavioral Survey. Colorado is highlighted in purple. The blue line represents middle school students, the purple line represents high school students. And interestingly, what we have seen over time is that marijuana use has actually remained fairly stable in Colorado high school students. And also we see that it is been not significantly different than that of the national, which is good news. Many people were concerned about youth obviously using marijuana once it became legalized in the state, but that is not what the data show here. And then with demographics in this age group, we see that the highest prevalence of past 30-day use among youth occur within those that identify as lesbian, gay, or bisexual, or trans. And then also use is most prevalent in the older grades, so the 11th and 12th graders. Marijuana method of use among high school students. So again, looking at how they're using smoking in this age group is most prevalent, but what we see and what is most concerning right now in Colorado is a extreme increase in the prevalence of dabbing. So again, the green line here showing a 52% of high schoolers who used marijuana in the past 30 days dabbed, and that is again using that highly concentrated marijuana. We don't know at this point if there's additional risk associated with that highly concentrated marijuana use, but definitely concerning to see this magnitude of an increase. So in summary for our youth, what we can say is that use has not changed among Colorado youth and remains similar to that, the prevalence of use nationally. Marijuana use is lower compared to other substances such as alcohol, and that changes, we are seeing changes in methods of use that are concerning to us. So looking now to the marijuana use and maternal and child health, looking to our PRAM survey, the Pregnant Risk Behavioral, PRAMS, just Pregnancy Risk Assessment Monitoring System. What we're looking at here is marijuana use compared over time over the years, and we're looking at use before pregnancy, during pregnancy, postpartum, and postpartum during breastfeeding. And so here again, we see pretty stable numbers. However, you can see again around the year 2014 when adult use became legal in the state, only about 11.2 of women were using before they got pregnant compared to 18.1 in the year 2020. So a significant increase there. But what we do see is that there has been really little increase over time among the those that are using during pregnancy and use postpartum and postpartum during breastfeeding. And these are important indicators for us, as we know, we have learned that THC can be passed to an infant through its mother's breast milk. So these figures we watch very closely, and we also see in the demographics of this population that the younger age groups, again, the 15 to 19 and 20 to 24-year-olds, the use is most prevalent during pregnancy and postpartum. And then we also see a correlation with the number of years of education or less education, we see more use in, and also among the population with unintended pregnancies, we see more use in. Okay, here we are. So looking now, this is the safe storage of marijuana in homes with children. So this is from the Child Health Survey. It's a callback to our adult BRFIS survey. And the question was looking at, do you store your marijuana in a home? If you have a child 1 to 14, how are you storing your marijuana in your home? And what we found out that the majority, actually, first of all, the homes with marijuana present, majority of homes in Colorado don't have, the adults are not using marijuana. So 86% indicated they do not have marijuana in their home. And then of those 14% who do, 89.6% indicated that they are storing their marijuana in a safe manner. So that is a good thing. And then of course, 10.4% in a manner that's potentially unsafe. And I should point out that in the survey, what's considered safe was the options were in a locked safe location, out of sight, but potentially unsafe was just like stored in a drawer, or in non-child resistant packaging. So there were some, again, this is a survey, so there were options. So then what we see with the outcomes in this population is that marijuana use during pregnancy and breastfeeding has remained stable in Colorado, and the majority of homes with children have their marijuana stored safely or is just not present. Now to our two other data sources that look at monitoring health effects or health incidents. So again, we look to Poison Center and our hospital data. There are some challenges with these data sources, particularly the hospital and emergency department data. They utilize the billing codes that are not their intention is not to be used for this purpose of monitoring marijuana use. And so that's a huge limitation of these data. We also saw a transition of the coding system itself in 2015. So not long after marijuana became, the dispensaries opened up in Colorado. So that became challenging to look at. Another challenging factor when we look at these data over time, especially around that time period. And then also we have to consider too that there could be reporting bias in these, i.e. you go to a hospital or emergency department, now that marijuana is legal, are doctors, nurses, intake, are they asking different questions on the intake? Are they specifically asking now, are you using marijuana, whereas before they didn't, which could cause an artificial inflation in these numbers that we're going to see. And then also these data do not speak to any long-term health effects. We're still kind of struggling to see if we can find a data source that can look at those and give us some information. So looking now at some emergency department discharge data, we saw a significant decrease for the second year in a row in 2020 among marijuana codes from ED discharges. And what we see, that is the purple line here. So it may not look very significant, but again, this is in rates per 100,000. So in 2019, it was 860.4 and then significantly dropped to 787.7 in 2020. So we see in comparison to other substances that marijuana is still significantly below alcohol up here in the green line compared to the stimulant and opioids also in the orange and yellow above there. Only one that's below marijuana is the cocaine codes. So looking now at the annual discharge rate of emergency department discharges with the marijuana poisoning codes. And what I should point out with these poisoning codes is that we've found that they tend to be more sensitive and specific when it comes to looking at these codes, meaning that we find it to be a little bit more accurate in looking at these data. So what we see here, and we're looking at, again, over time, rates per 100,000 in different age groups. What we saw between 2019 and 2020 is significant decreases in different populations, but we did see a significant increase in our younger populations. So in our 0 to 9-year-olds, and then also in our 0 to 8-year-olds, and then 9 to 17-year-olds, we saw increases among those, which is concerning as well. We obviously don't, want that age group to be consuming marijuana, and much less be going to the emergency department. Looking at poison center now, poison center data, we look at the annual frequency of marijuana reported exposures, and we're comparing those here to alcohol. So alcohol is up again in the green line up top. The purple and blue represent the total marijuana exposures. The purple is marijuana only, blue is marijuana plus other substances involved. And what we see here is, again, you see with the opening of dispensaries in 2014, a huge jump in the number of reported exposures to the poison center, which remained fairly stable up until about 2018, when we again see like another increase. Not sure why we see that there, but it is something that we're keeping an eye on, and we're seeing continual increases in people calling poison center. Looking more closely at the demographics of those calls, what I'd like to point out here again is the age groups which these exposures are happening in. The red line here indicates the 0 to 5-year-old age group. You can see that accounts for the majority of calls since 2017, roughly. And then of those, the intentionality of the exposure, so was the marijuana use intentional or not, indicated by the red line below. We also see that the majority were not intentional. So in summary, we're seeing increases in emergency department visits among those younger age groups with the marijuana related billing codes. We're seeing decreases in marijuana related poisoning billing codes. And then we're also seeing reported exposures highest among the younger ages and unintentional exposures. So now moving on to our emerging science and medical information. So this is the second part of the statute that we're required to do. I'm going to talk to you a little bit about our advisory committee, which is an appointed panel of scientists and professionals that help us to monitor our emerging health effects in the literature. And I'll go into that. So first, here's our committee. This is their areas of expertise that's represented. As you can see, we have quite a spread of experts. And we cover these 11 topics. Last check, I think we were at like 33 or 34 subcategories for these. So we do look at a big breadth of information that we conduct our literature reviews on. And so the goal of all this is to translate the science into public health. We develop evidence statements that convey the quality and quantity of the evidence behind a finding. So we have evidence statements that reflect the science directly. We translate those into public health statements, and then those help guide and develop our prevention campaigns. And so here's just an image, a visualization to help put into perspective what I just said. So what our evidence statements are, they're more scientific. They take, all the articles that we read on a topic, they weigh them, and then we were able to come to consensus with our advisory committee on a very scientific type evidence statement, which is there on the left, where beginning with biological evidence shows. We then try to whittle it down to a 10th grade level of language for our public health statements. And then our partners in our prevention services, part of the health department, then whittle it down further to a 7th grade level, which then is used in our prevention and education campaigns. So that covered our assessment, and what we'll go into next is the policy development. So speaking of our prevention and education program here at the health department, they work to create public awareness and education on the laws. So that was one of their first tasks that they were issued back in 2014, was to educate the public on where you can use safe storage, and what later came to light was safety on edibles, since we later discovered that we had a number of high publicity incidents in Colorado in which edibles were consumed, and we didn't realize at first that they took a while to kick in. Unlike when you smoke marijuana, it's immediate. So a lot of education and awareness went into those, into that. And so then prevention use among youth and pregnant and breastfeeding women, that's another very highly important population. Again, as I mentioned earlier, with the THC passing to an infant through breast milk. So there's dangers of use, potential health effects and consequences. And we've had a number of campaigns over the year. The one on the right, Good to Know Colorado, was one of the first ones. Our current one is the cannabis.colorado.gov. It features the Responsibility Grows Here campaign. And then finally, we also have an occupational health and safety education report that we put out a few years ago, specific to those working in the marijuana industry. Partnerships is another piece of this, part of the public health framework. As you can see on the right, we have formed many partnerships with other state agencies. We all work together to help inform policy. We also work with our universities, our local public health agencies, and stakeholders on both ends of the spectrum, as well as some national partners as well. So in policy development, we've learned from past experiences over the past 50 years of what worked well and what was successful. So we took some of those lessons learned and implemented them in Colorado in relation to cannabis legalization. So of first on there, the age 21 plus strict age limit to consume retail marijuana or to buy retail marijuana, limited hours of operation, a higher tax rate, These are all evidence-based strategies that we implemented. And then we've also, as I mentioned earlier, at the health, as part of our health department role, we do consultation on regulations. So this is also seen reflected in the policy with the density of dispensaries and location of dispensaries, the policies regarding packaging, and then most recently, we've now have limits on the amount of concentrate, marijuana concentrate, that can be purchased in a given day. So moving on to the very last third section of the public health framework, assurance. The enforcement piece of this, as I mentioned previously, we have the Marijuana Enforcement Division that works at it's our regulatory agency. They work out of our Department of Revenue. They are the entity that's responsible for the tracking, the sales, the inspections, and adverse health events are required now in rule to be reported to them, which we are partnering with them to help us document those and get a good data source out of those. The enforcement also applies to product safety. We are also heavily involved with that as well on the lab testing front, helping them develop rules and regulations regarding what's allowable as far as those contaminants listed there. But there has been challenges along the way, especially within that laboratory piece, determining the limits and new products that have come on the market in the recent years. The other piece, assuring a competent workforce. We have, again, worked with our Marijuana Enforcement Division on a responsible vendor program. This is aimed to educate those who work in the industry on health effects, safe and responsible use, and their legal responsibilities. We also, again, as I mentioned previously, our worker health and safety report that we put out. The biggest challenge with this piece of assuring a competent workforce is that none of it is actually required by law. So the responsible vendor program is completely voluntary at this point in time. So that may be changing, but as of right now, not required. The evaluation piece, what we do here, obviously we're collecting data. We're always looking for new sources of data to help us tell this story more clearly and more accurately. As I mentioned earlier, there are a number of limitations to the data that we do have, and this impacts the way that we can interpret that and apply it to the state of Colorado as a whole. So, we're really limited in what can and cannot be said based on those limitations of the data. Comparison with other validated measures. So as I showed you in some of our data, we look at Colorado compared to national. And what we hope to start doing too is as we get more on board with some of our other partners, both federally and nationally, other states, we would like to be able to compare data, especially from those other legalized states as well. But it's difficult right now because a lot of us don't have the same methodology or questions in order to do an apples to apples comparison. So efficacy of prevention messaging also, we need to consider what's culturally appropriate. I will throw out the ongoing debate right now of using the term, cannabis versus marijuana. One that's an issue because it has different meanings, different to different populations of people. So that's just one thing that we're working on evaluating and trying to figure out better. And then what resonates, what prevention messaging resonates with certain populations the best. So I use the example here of teens versus pregnant mothers. So, what might resonate with those teens isn't going to resonate with all pregnant mothers. So we have to be very conscious and keep on evaluating our techniques and our prevention messaging. The last piece of the wheel here is the research. We do have a marijuana research grants program, which I oversee, that was also established in 2014. The story of this is as we go through our research and our literature review, we're constantly identifying areas that there's large research gaps, not only in retail or adult use, public health effects, but also in the medical field, the efficacy of whether or not marijuana could be used to treat a certain condition. And so this research piece really gets to the heart of the issue. We need more data, we need more studies performed, and good studies performed to inform our information and our decisions and policy going forward. So overall challenges, these are really ongoing themes that have been going on now for quite a while. Some of them are newer, but what can be done to improve public health policy, right? All action, requires supporting data. And as I said, we're always looking for more data, better data to help tell the story more clearly and accurately. One of the newer themes, as I said also, is the issue with high THC products, those concentrate products that our data show that the youth are using to dab. Very concerning. We don't know, again, if they're more or less hazardous to health. Preventing unintentional ingestion by children, as you saw by our poison center and hospital data. The younger age groups are accessing and getting exposed to marijuana. We do have childproof packaging in place, but that's clearly not enough at this point. So what can we do to further the prevention of that? The list can go on, and also some of these are definitely not unique to Colorado, such as testing for impairment and DUI. And then we're also seeing more social consumption policies take place, not only in Colorado, but I believe also in Nevada as well. So with the overall public health impact here, what we are showing, with our surveillance and our scientific review, is all being disseminated through our reports and through our website. And we are hoping to, collaborate more with our federal, state, and local partners, as well as our stakeholders to get this information out. This presentation also is a perfect example of how we hope to share our information for others to utilize. And here is our webpage where you can view all of our data and all of our evidence statements. Our web address is marijuanahealthinfo.colorado.gov. So we hope you take a visit there. And I thank you for your time. And I'm sorry that I didn't have my slides up correctly at first, but thank you.
Oh, that was a marvelous presentation. You're very well organized and have facts at your fingertips. That's look ahead for states like North Carolina that are just considering this now. So I see there are some questions have come in. Let me just read them. Robin Huffman said, "How much revenue is produced through the taxation?"
Quite a bit. We've seen over time a continual increase. I'm sorry I don't have exact figures for you. Those are data from my partners at the Department of Revenue, but I can tell you we have seen over time just an increase every single year of tax revenue collected. So I can what I can do is I can see if I can find the link to that and put it in the chat, or follow up with Dr. Levin to give you that information.
Okay, and I can distribute that, yeah. And Nick Tice said he's read that there are some efforts to ban the dabbing pens in Colorado. Is that right?
Yes, so the dabbing, there's different ways you can dab, but essentially what last legislative session, there was legislation introduced that was trying to limit, or to limit the percentage of THC in marijuana. So it started out very big. It was a very wide cast net. The issue at hand were those highly concentrated products, right, which can go up to upwards of 80 to 90% THC. However, the legislation, the way it was written, was trying to limit to a certain lower percentage, like 10 or 15%, which would encompass not only those particular products like the waxes and the oils, but it would also encompass the flour that was available as well, since a lot of flour tends to be over 15% THC. So everything kind of went back to the drawing board. What ended up, being passed was something completely different, and not limiting any particular products. So as of right now, there are no product moratoriums or banishments in place. So what we're seeing, though, is just an ask for more research and recommendations on those high potency or high percentage THC products before any further policy is recommended from there.
Okay, and there was a request. Could you please place the link in the of the website reference for the data in the chat?
I will do so.
Okay, thanks. And I just had a question. We had this discussion earlier about with legalization, that one possible benefit would be a regulation of product quality. And there's the comment that they've discovered mold in a lot of the samples of cannabis and other things like pesticides. I mean, is there an effort in terms of maintaining the quality of the product that's sold through the dispensaries?
Yes, so part of our cannabis sciences program, which I'm part of at the State Health Department, we are based out of the state lab. My teammates are part of that group who makes the recommendations, the reference library of sorts. So what they do is they not only validate the laboratory methods on how to test for whatever kind of contaminant it is, be it metals, mold, or anything of those sorts. So they not only develop those, but they also consult with our regulatory agency, the MED, the Marijuana Enforcement Division, on what limits to set in rule about that, right? Which is product safety in essence. So, whether it be, less than or equal to 10 parts per billion of something, that's all, informed scientifically through stakeholder engagement, I should mention as well, but also through consultation through us at the State Health Department Public Health Lab.
Thanks. Any other comments or questions from the panelists? Or?
Yes, I do. So several years ago, and by that I mean 2015, there was some data that was produced by, I think, the health department that specifically speak to regular marijuana use, lots of marijuana use, and it actually mentioned half of a joint or a whole joint in terms of outcomes. And so I don't think I heard in your presentation, forgive me if I missed it, that spoke to the regular use of marijuana possibly leading to schizophrenia and bipolar in young adolescents and young adults. So is that still something that's published by the Department of Public Health?
It sure is, and that is my team. And thank you for such a wonderful question, and thank you for all the other questions as well I failed to mention. But yes, so there is, our advisory committee, we have an evidence statement that speaks to exactly to what you're talking about. And we say that there is substantial evidence that shows that, the earlier the use and also the more frequent use, so daily or near daily use, in the younger age group is strongly associated with mental health, with a mental disorder later in life, such as schizophrenia. We do have several statements about that on our website, and I did post the link in the chat for everyone. So if you go to the literature review tab and then click on evidence statements, you can sort by population there, and that one that you're speaking to is under our adolescent category. But you also bring up a very good another problem, right? So we not only do we look to categorize and describe the frequency of use, but one thing that's so challenging that no one has really been able to get a grip on is how do you describe the amount of marijuana being used, the quantity. Not only do you have different varying levels of THC in these products, but then you don't know the actual amount that person is inhaling, or that they're going to dab, or I think that the one exception to this is the edibles, in that exactly how much you're getting per cookie or per piece of chocolate. That is the only way that we've been able to really quantify marijuana use within those edible products. So it's still a lot of challenging work ahead of us, even 8 years into this.
Yeah, and previously the definition was heavy marijuana use daily or near daily, either a whole joint or a part of a joint, specifically calling it a joint, 5 to 7 days per week, regular marijuana use weekly, 1 to 4 days a week, a whole joint or part of a joint, and then occasional marijuana use less than weekly, part of a joint or a whole joint. So, perhaps that's changed. But then is there a lot more attention and cost that's associated with mental illness in Colorado because of these statistics? Is a lot more money going to treatment and whatever else one can do in terms of mental illness in Colorado?
Yeah, that's a very, very good question. And, mental health data are always difficult to come by because of, the federal guards around them. The mental health data that would probably best answer that question, our health department doesn't collect. It's actually another state agency in Colorado, the Department of Human Services, Office of Behavioral Health. So I'm actually not familiar with that cost, but what I can speak to is that a lot of, and I'd have to look this up again with the other revenue question, but I know a good chunk of the tax revenue generated from the sales of marijuana does go to, behavioral health, mental health treatment. So, and that's not just specific to marijuana, that's for all substance abuse. So I'll have to circle back with that one as well, as far as, but yeah, as far as like cost to treat mental health, I don't know, unfortunately.
Thank you.
The two big players in terms of intoxicating substances are alcohol and marijuana. So have you noted any change in alcohol consumption since Colorado's had legalization of marijuana?
Well, our data indicate, it looks to have remained fairly stable throughout this. And I don't think we unfortunately just lost our alcohol epidemiologist a year ago. Yeah, she got reassigned to COVID and we haven't seen her since. So, unfortunately, I don't have the exact figures on that. But it's also an interesting question as well. But from what I can tell from our data, it doesn't. Let me just take a quick look at it real quick. Is there another question while we look this up?
Let's see. It seems that like a naive question on my part, but once a state establishes retail marijuana sales, did that eliminate the need for patient registry and separation of medical marijuana? So did you have a medical marijuana designation when it was just medical marijuana, and then did that kind of evaporate when you went all the way to recreational use as well?
Yeah, that's a very good question as well. So we, interestingly, what we saw was there was, I wish I had the slide to show you this, but we had medical marijuana since 2000, and the number of registered card holders remained very, very stable up until around 2009. And all of a sudden, we saw at the state health department, our medical marijuana registry saw a huge surge in the number of people applying for their medical marijuana card. I mean, it went from like, I want to say 18,000 to like almost 100,000 in 1 year. So exponential increase in the number of cards. And timing wise, that happened right before the commercialization of medical marijuana occurred in 2009, 2010. So we believe that with that, knowing that the commercialization was coming, that a lot of people then decided they wanted to get their card. And so that's why we saw the surge. And then once retail marijuana or adult use marijuana became legalized in 2012, we were expecting to see a drop in the number of people wanting to get their card, but we saw a very small drop, and it remained very stable for a long time. And so what I think that speaks to more is the need for the stores for our medical patients, rather than the need for or the want for recreational use, if that makes sense.
Yeah, sure does. Okay. Are there other questions out there? Not seeing them. I really appreciate your coming and speaking with us. And if other questions come up, is it okay if people email you?
Sure. I think Diana has a question.
Diana has a question. Okay.
Yes. Hi. Thanks so much for your presentation. I really appreciate all the efforts of Colorado for generating these tables and doing the statistics. It's beyond, it's wonderful. None of the other states do it. We really appreciate it. But my question is, dispensaries and the information that dispensaries send out, and warning labels. I know there's warning labels on the product, but I know there's a study that surveyed the dispensaries and the information that they gave pregnant women in terms of what the effects of marijuana were on morning sickness, and many of these dispensaries, 70% of them, I think it was, suggested using marijuana for morning sickness, and virtually a very small percentage said consult your health professional. So is there any kind of effort to fix that? Because of course we know the dangers of marijuana in pregnancy.
Yes, and yeah, that one dreaded study, right? We all frown upon in public health. What's unfortunate about that is that the rules and regulations have long been in place that say anyone working at a dispensary or a store is not allowed to make recommendations. So I should also specify in Colorado, when you are a medical a doctor treating a patient and using medical marijuana, you are not prescribing it, you are recommending it.
Oh.
And so that word recommendation comes very importantly into play here, because in rules and regulations, no dispensary employee can recommend marijuana for anything, any specific medical condition. And so that has long been in the rules. And this is a case of unfortunate, under I guess, enforcement. I mean, it was one thing we didn't know it was happening. And second, are there, if you don't know if it's happening, how do you stop it? But yes, we have since that report came out, our partners at the regulatory agency have, since done some education to the marijuana industry on you and your employees are not allowed to recommend this. And one of our stakeholder groups in particular also took it upon themselves to create signs that dispensaries could voluntarily hang up that say, marijuana is shouldn't be, consumed by pregnant or breastfeeding women. They basically took our statements from our advisory committee and turned it into a sign that could be hung in a dispensary. There's, question as to whether that's an effective prevention measure or not, but yeah, that's one piece of action that was taking place.
That's good. So I had a question. So furthering along this kind of permission line, so it's legal and available in Colorado, but is still illegal federally. So do you or do the people in dispensaries or in the industry or individuals run into any problems with that disconnect between the federal laws and the state laws?
We have not, as far as the public health realm. The enforcement, I think there may have been, I don't think there's been any issues, but I think there's been more like cooperation and collaboration, especially in investigating perhaps illegal grow sites in the state. But, I can speak there to some issues that have happened on the research front, especially conducting random controlled trials using marijuana, as a treatment. Initially, when we started the research grant program in 2014, some of our researchers that we grant funded ran into some unforeseen obstacles getting their licenses and securing product and such. One in particular unforeseen obstacle was actually conducting the research using a Schedule I substance on a university campus that gets federal funds. So that one was a tricky one to navigate. So I think in the research realm is really where we've seen the biggest obstacles.
Yeah. I mean, I actually have a Schedule I license, so and I appreciate that. There's another comment here, a question about from Robin Huffman. North Carolina, we have this kind of almost a sting operation, I guess, secret shopping, where you have youth go in and try to buy cigarettes and who are clearly underage, and just to see if the retailers are obeying the law. Is there a similar thing with the dispensaries, or? Yeah. Yes,
our marijuana enforcement division actually conducts these sting type operations with youth. I don't know how frequently they do it, but they do do it, and I think I'm happy to report that I think majority of dispensaries, like vast majority, don't sell. So yeah.
Good news. Okay. Well, I want to thank you for taking your time out, and it's a marvelous program you have there, and I look forward to hearing more about it in the future. Okay.
Thank you all very much. Thank you for the wonderful questions and the opportunity to present here today. So thank you.
Thanks. Appreciate it. Okay. So now, Wanda, did you have some further remarks that you want to kind of have some discussion?
Well, I think we're going to shift now to just a general discussion. Anyone that would like to have something else to say, I will say on behalf of TRI and, oh, Nicole has her hand up. Thank you. That our prevention efforts include alcohol, tobacco, marijuana, prescription drugs, vaping, and we use 7 strategies for community change in the public health model. So after we're finished here, please get in touch with us. And this is not a goodbye. It is just a public health announcement. Okay, Nicole, go ahead.
I was wondering if we could hear, we got a little crunch for time earlier in the day from Dr. Dow-Edwards and Dr. Parnell, that, your talks were very scientific and really deep into the latest science. But if you were talking to your, a niece or nephew or a child, what would be your takeaway for any of your family members about what does your research say about what our societal response should be to marijuana?
You want me to go first? I can go first. Off the cuff, it's difficult. I think let's focus on adolescence for a while. I have a whole, pregnancy is a whole different issue. But in terms of adolescence, I know there's peer pressure. And of course, the old reefer madness idea that if you smoke one joint, you're going to be psychotic forever and all this stuff is just not realistic. It's not true. But if you look at the literature and you look at the controlled trials, the published studies where they have done things in a non-emotional scientific way, the dangers of the drug THC by itself is quite remarkable, quite long-lasting for adolescents and different from the adult. The adult effects on memory and learning and so forth do become normalized, and many of the effects in the adolescent do also get less, but the overall there is a long-term detriment to brain function. And it's seen in many, many papers. Unfortunately, people get very emotional with this. I've given talks and I've had people come up to me and say, I'm wrong, and yada, yada, yada. And I'm just giving the animal data. I'm not telling anybody what they should or should not do. So people do get sort of emotional and they are not really listening to the facts. So what I would say, I have 2 sons. They don't really ask for my advice ever. But if they would, I would say, everybody's going to try it. You're not going to drop dead if you try it. But harm reduction, that whole idea of use less potent routes of administration, the THC CBD ratio is very important. Particularly, all the data supports that, even particularly with psychosis. CBD alone, unfortunately, is really not adequately studied, and yet you can get it everywhere, But certainly the data along with THC supports the fact that, something with a more favorable, meaning more CBD ratio would be safer. Staying away from edibles that have high THC concentrations, staying away from vaping, which also has high THC concentrations, and that it is dose related. The earlier you start smoking, the more potent the marijuana that you smoke, the bigger the effects. And that's what I would say.
Scott?
My approach is somewhat similar. And that one, I appreciate what Diana was just talking about, people being emotional. Now try throwing pregnancy and newborn baby into that mix. What I tell people is generally is the same that I would tell them for alcohol. Yes, smoking a joint is not going to most likely kill your baby, just like having a glass of wine is not going to cause severe birth defects, but it is a cumulative dose response effect, right? And when people ask me about alcohol, they ask me, well, what is the safest limit? And I tell them, we don't know. It depends on your nutritional factors. It depends on genetic factors. My lab does a lot of alcohol genetics. There are big genetic components to these things, as there are with any disease. So, and like Diana was just talking about, it also depends on potency, how much THC you have. Now, I will say that for early development, I don't think that having more CBD in there will help, because CBD is also a sonic hedgehog pathway antagonist. So both THC and CBD are inhibiting sonic hedgehog pathway. And while CBD is will antagonize some of the effects of THC at the CB1 receptor, you're still getting a net overall inhibition of this really critical growth factor. And I, like Diana just said, we need more research on this. We need, yes, we have replicated, well, my collaborators have replicated what we have done in the mice with the fish. But we need more work. We need more recent work in humans. All of the data out there the majority of the data out there is from people who were born in the 70s and 80s, when marijuana was not used nearly as much as it is today, and when the THC concentrations were so much lower. So I guess that's what I would say.
I agree with your comment about CBD. Just because it's not intoxicating doesn't mean it's not toxic. I mean, exactly. DDT doesn't make you high, but it's quite toxic. Right. There's just very, very little information about it, and it's amazing how widespread its use is become without data to back up safety or efficacy. So.
Yeah, especially since it's being used or it's being touted as being effective for morning sickness, which it seems to be efficacious for that. Unfortunately, people tend to get morning sickness right around these early critical periods of brain development. Yeah. Not that any period of brain development is not critical, but the early time points that we were looking at, that's usually when people get morning sickness.
Thalidomide was originally used for morning sickness.
Right.
So anyway, there are a couple of comments here. Stephen Wyatt's been very patient because they've been up here since about 12:30 or so, pointing out, rightly so, that there are substantial individual differences in terms of susceptibility to adverse effects or maybe other effects as well of THC. So something to be on guard against. And like with any drug or any chemical, some people may be very susceptible and other people quite resistant. So as the research goes on, I think this needs to be attended to. And then he had another comment about the significant peer pressure that goes on in terms of its use and strategies in terms of addressing that. So that harkens back to the speakers and the panels that we had in terms of adolescent use. And then one from Nick Tice about the vape shops in North Carolina selling dab pens, THC. Maybe Delta 8, which technically I guess is not illegal, but like with a lot of things, like with the amphetamines, that there's this kind of progression of new synthetic analogs that keep one step ahead of the law. And I think that goes with cannabinoids as well. So maybe that's another possible benefit of legalization of the traditional Delta-9 THC, is that then there's kind of puts a stop to that sort of whack-a-mole. So, make this illegal, and then that comes up, and, unknown risks or potency. So again, any other comments, questions? I think this has gone extremely well. I got an email comment from one of our faculty members here, Cindy Kuhn, saying it's very rare that you see this sort of collaboration with community members and people doing research and having a kind of full spectrum of voices heard in terms of impacts of something like marijuana or anything else, in terms of things in the laboratory, things in the clinic, things in the schools, things in the community. A lot of these are very cross-cutting, and it really, I think, is to everybody's benefit to bring all these voices together. And so I wanted to thank everybody, Tyler, for an outstanding job juggling the technology and bringing this all together, and somebody's on and somebody's not on. It's like trying to make everything work, and Nicole for her key part in putting this together, and in particular Wanda for being a wonderful collaborator with all of this, and all of our speakers and panel members. So wish you well, and keep in touch. If you go to the CABC website in a few weeks, you'll be able to access the video version of this, and it'll take a little while to get edited and so forth, but we'll have that archive. And also we have a regular seminar series. Linda Perino is going to be presenting. She's from Wake Forest, on her work with drug abuse research over the last few decades. That'll be next Wednesday. So just go on the CABC website, just Google Duke CABC, you'll come to the website and you can clock in from there. Okay, well, with that.
And so I wanted to thank everyone as well, the participants and the panel, and CABC for participating together with us in our annual conference. We look forward to having this collaboration again next year for year 3, but celebrating our 21 years. If you go to, thank you, tryforresilience.org. That is our website, where you'll see all of the different strategies that we use for our prevention efforts. And so again, just as Ed said thank you, I say thank you too, and have a marvelous rest of the day.